So, hello students, welcome back to Vedantu NEET English. This is 𝐂𝐡𝐞𝐤 your biology master teacher. Quickly, let me know in the chat if I am audible and the presentation behind me is visible to all of you, because it's very important, right? Because if I'm not audible and visible, what is the point, right? So students, this is the part 2 of biomolecules. Part 2 of your biomolecules. In the first part, right, in the first part, we finished something called as the experiments to analyze the chemical nature, that is living, or that is organic and inorganic materials. Then we did something called as the different types of amino acids. Now we studied about structure of amino acid. We also learned about acidic, basic and neutral amino acids. We also learned about something called as zwitterion. We learned something called as essential and non-essential amino acids. So all that was covered in previous class. And also we did something called as your lipids, right? In lipids we learned about fatty acids, the structure of fatty acids, the structure of glycerol. And when we add glycerol and fatty acids together, we get glyceride. That also we learned in yesterday's class. We also finished off the class with something called as primary and secondary metabolites. Right? So students, that was the first part of biomolecules. So if you have not watched the first part, I would highly advise, I would ask all of you to watch the first part before watching the second part, because that is also very important, right? So nevertheless students, quickly smash that like button. Quickly smash that like button, all of you, and mark your attendance. And all the students who are watching it recorded, all of you also mark your attendance quickly, all of you. Now students, what will we be doing in today's class then? In today's class, what are we going to study, right? So we'll be starting off with something called as polysaccharides, right? We'll be starting off something called as macromolecules, polysaccharides, where we'll be studying something called as different types of carbohydrates, right? Different types of carbohydrates basically, right? Different type of carbohydrates. Now after we finish carbohydrates, tell me in the chat, all of you, how many of you face difficulty in the enzyme part? Don't be shy, raise your hands, all of you, in the chat. Hari, right? Pala, Harika, Chotu, Abhinaya, all of you, Gracie, raise your hands, all of you. How many of you face difficulty with the enzyme part? Madhumiti, right? Madhumati. So tell me, do you know the difference between Vmax, half Vmax, Km value? What is this called as inhibitors? What is this enzyme complex substrate? What is this what is the transition state? What is going to be competitive, uncompetitive and non-competitive inhibitors, right? So if you have any doubt, regarding any of the parts, right, today is the day that is going to get cleared. I am very sure after reading the lines of NCERT one by one, you will confidently tell me, sir, yes sir, I am confident in this part. I can solve any question, including even cofactor also. What is this prosthetic group? What is this metal ion? What is this organic, different types of catalyst? So all that, all the information, the crucial information of biomolecules we'll study in today's class. So let me know in the chat if you're excited for today's class. Let me know in the chat if you're ready for today's class, because it's going to be a power pack class. Ready, all of you? That's easy, yeah? Amazing. You all raising hands. Yes, raise your hands. All of you, raise your hands quickly. Raise your hands. Don't be shy. Who are facing difficulty in enzyme part? It is very easy. It is very easy. It is a laddu concept. Okay, it's a very laddu concept. After I teach you, just let me know how you want to how you feel about those concepts in the comment section later on. So with that being said, let's start the class. Now students, before we read, can anyone tell me what is the meaning of carbohydrates? The name only tells carbohydrates. They are what? They are hydrates, right? They're basically hydrates of carbon. They're basically hydrates of carbon. Now if you look at the different structure of your carbohydrate, can you notice, all of you, can you notice different OH groups everywhere? There is carbon and there is hydrogen chains. Carbon hydrogen chains, yes. So there are hydrates of carbon. That is basically your carbohydrate. And students, if you look at a carbohydrate, can you notice this part here? In the case of your glucose, we have an aldehyde group, and in the case of your fructose, we have a ketone group. Yes. So can I tell? Usually they consist of a aldehyde group. Aldehyde group, yes. They have aldehyde group, yes. they have a aldehyde group or they can have a ketone group. They can have aldehyde or a ketone group. Aldehyde or a ketone group. All of you will agree with me. Now students, there is a small classification here. Now you tell me, all of you, use your simple logic. What is the meaning of mono? Tell me, what is the meaning of mono? Mono. Mono means one. Okay. What is the meaning of di? Di means two. What is the meaning of oligo? Oligo means 3 to 4. What is the meaning of poly? Poly means many. Many. Okay. Many. Now students, if I add these many types of monomers and if I add a saccharide to it, that becomes a simple classification. For example, if you look at the different classification, we have monosaccharides. Mono means one, saccharide means sugar. A single sugar is present, that is called as a monosaccharide. Di means what? Di means two. Two sugars are present together, they are bound together, that is called as a disaccharide. Oligo means 3 to 4. 3 to 4 sugars are joined together, that is called as an oligosaccharide. And poly means many. Poly means many, that is many sugars are joined together, that is called as polysaccharide. Polysaccharide. Now students, if I ask you, can you tell me, apart from that one line, can you tell me something more about monosaccharide? Monosaccharide. They'll ask you, students, you need to understand, all of you know, the first point here is what? About monosaccharide, it is the simplest carbohydrate, right? It is the simplest carbohydrate. Simplest carbohydrate. Now, since it is the simplest carbohydrate, can you reduce it even more? Tell me, yes or no? It is already the simplest carbohydrate. Can you even reduce it more? Can you hydrolyze it to something even more simpler? No. You cannot. So can I tell it cannot be hydrolyzed further? It cannot be hydrolyzed. Cannot be hydrolyzed further. Cannot be hydrolyzed further. It is the most simplest form of carbohydrate, a monosaccharide. A monosaccharide. Now students, quickly tell me an example of a disaccharide. How many of you know an example of disaccharide? Yes. Disaccharide. Tell me an example of a disaccharide. A simple example of a disaccharide. Any one of you? It's not given anywhere, no? Not given anywhere. Okay. Good. I thought it was given anywhere. A quick example of disaccharide, all of you. Any example of disaccharide? Okay. Any example of a disaccharide? Anyone? Quickly go in the chat. I'll give you a simple example. Friends, how many of you like chocolates? How many of you love chocolates? Sweet chocolates. Sweet chocolates, right? Can I tell the scientific name of this chocolate, or just saying, can I call it sucrose? Your sucrose. Your sucrose is an example of a disaccharide. Now I told you the meaning of di is what? Meaning of di is 2. So this sucrose should be made up of 2 different sugars. It should be made up of 2 different sugars. Now your particular sucrose here is made up of, sucrose is made up of glucose plus fructose. Glucose plus fructose. That is your sucrose. Okay. Now tell me, how many of you like drinking Boost, Bournvita, or Horlicks, or tea, or coffee, or any kind of hot beverage, like any kind of hot drink? You will tell me, sir, I love milk, right? I love milk. So can I write other example as lactose here? Lactose. Now what is lactose made up of? What type of 2 sugars is lactose made up of? Anyone in the chat? Yes? Alpha beta glucose in chemistry, not here. Chemistry you learn. Diksha ma'am will teach you in chemistry what is alpha, what is beta, the linkages, right? The alpha linkage, beta linkage, all that in chemistry you will learn, not in my class. Mine is very simple. Okay. This lactose is made up of glucose plus galactose. Glucose plus galactose. Now you might be wondering, sir, I am writing plus anywhere. I am writing plus everywhere. Okay. Now what is the bond, students? What is the bond which is present between these two? You may be wondering, sir, why I am writing plus here? Obviously there should be some kind of bond there. You cannot make one, you can't use Fevikwik and add Fevikwik and join two. Obviously you can't use Fevikwik. There has to be some form of bond, the friendship bond, no? There should be some friendship bond between glucose and galactose or glucose or fructose. What is that bond called as? What is that bond called as? Anyone? Anyone? What is that bond called as? Glycosidic something? Remember something similar to glyco? I'll give you a small clue. Glycosidic, exactly. Kaushik, it is called as glycosidic bond. The bond here is called as glycosidic bond. Beautiful, amazing, amazing. All of you are very smart today. Extra smart you became. Okay, extra smart today. Glycosidic bond. Glycosidic bond. Yes, glycosidic bond. Okay, glycosidic bond. Now that is your disaccharide. Now can you give me an example of oligosaccharide? Tell me how many of you know oligosaccharide? Any example of oligosaccharide? How many of you heard about raffinose? Raffinose. Raffinose is a type of oligosaccharide that they will not give an examination. Don't worry, that they will not ask you an examination. Raffinose and all. So this much all of you understood, tell me. This much all of you understood? Okay, nice, nice. Now we will understand something called as polysaccharide. Polysaccharide. Okay, polysaccharide. Tell me students, is polysaccharide made up of a single monomeric unit or more than 10 monomeric units? Quickly in the chat, all of you. It's a polymer of monosaccharide. It is a polymer of monosaccharide with more than 10 monomeric units. Monomeric units. More than 10 monomeric units. Now, more than 10 monomeric units, I told you. Now, students, understand this one simple thing. Do all of you love Kit Kat? Some of you will tell, sir, I only like Kit Kat I do not like Munch or anything. But some of you will be like, no, sir, in the morning I will eat Dairy Milk, in the night I will eat Kit Kat. So students, just like how you have preference, your polysaccharide can also be divided into two categories. There is one group which has similar type of structure. Like some of you like only Kit Kat, right? So there is some group which has only one type of sugar. They will only like Kit Kat. There is one group which likes multiple types. I call them hetero. So students, here we have something called as homopolymer. We have something called as homopolymer and heteropolymer. Homopolymer and heteropolymer. Homopolymer and heteropolymer. Homopolymer as well as heteropolymer. Heteropolymer. Or I can write homopolysaccharide. Let's write homopolysaccharide. You'll get confused otherwise. You're telling what I'm telling. Homopolysaccharide and heteropolysaccharide. Polysaccharide and heteropolysaccharide. Polysaccharide. Now students, this homopolysaccharide, right? This homopolysaccharide can again be further divided. All of you know Kit Kat has so many flavors. All of you know Kit Kat has so many flavors, right? If you go to Japan, you will know so many beautiful flavors are there. Okay. Now here, your homopolysaccharide can again be further classified into two categories based on structure and based on storage. Based on structure and based on storage. Based on structure and based on storage. Structure as well as the storage. Now, based on structure, students, based on structure, we have something called as cellulose and chitin. Cellulose and chitin. Cellulose as well as chitin. Okay. Cellulose you find in the case of plants. Chitin you find in the case of your insects. Now you'll ask, how many of you like cockroach? Like cockroach, you know, you'll find chitin. Okay, you will find chitin. Now here, in terms of storage, we find three different categories. Okay, there is something called a starch, which is the storage material in the case of plants. We have something called as inulin. Inulin is also storage material in the case of plants. Then we have something called as glycogen, which is the storage food material in the case of animals. So we have under homopolysaccharide structural cellulose and chitin. Under storage we have starch, inulin and peptidoglycan. Any doubt there? Quickly ask me. Any doubt there? Very simple, no? Very simple. Now under heteropolysaccharide we only have two examples. Very simple to remember. I'll give you a clue. Okay. The first one is the material present in the bacterial cell wall. Yes, bacterial cell wall. Can anyone tell me? In bacterial cell wall we find it. Like, you know, bacteria has three layers. Yeah, right, all of you remember, right? Bacteria has three different layers. Outer layer, glycocalyx. Glycocalyx can be present in the form of two forms: slime layer and capsule. Second layer is called as cell wall. Third layer is called as plasma membrane. All of you remember that. Three different layers of your bacterial cell wall. Now this middle wall, cell wall, is made up of something called as peptidoglycan. It is made up of something called as peptidoglycan. This peptidoglycan right here is made up of NAG and NAM. N-acetyl glucosamine, N-acetyl muramic acid. Okay. Now students, the second one, I'll give you a small clue. You should tell me in the chat, or if you're watching it recorded, try guessing it after my clue. The second example which I'm trying to tell you here is, it is obtained from algae, gelidium. Okay. It can be used in jellies. It can be used in microbiology experiments to make the solid media. Yes. Anyone who can tell me now? Remind me again quickly. The second which I'm trying to tell you here starts from A. It is used in jellies, making ice creams. You have studied about this in the plant kingdom chapter. It is the structure which is present, which you use in your microbiology lab. Anyone? Quickly. Agar. Exactly, Ravindrani. It is the agar, which is an example of heteropolysaccharide. Let me quickly revise for all of you students what we learned till now. We learned something called as carbohydrates. They are hydrates of carbon. They can have aldehyde or ketone. The classification is very simple: mono, di, oligo, poly. In monosaccharides, simplest carbohydrates cannot be hydrolyzed further. Disaccharides, sucrose, glucose plus fructose, lactose, glucose plus galactose. The bond here is glycosidic bond. Then polysaccharides, we have two types. All of you, two types of polysaccharides. We have homopolysaccharides. Homopolysaccharides will have what? Only one type of sugar. In homopolysaccharide. For example, in chitin, cellulose, starch, inulin, glycogen, we only have one type of sugar, that is mostly glucose, except in the case of inulin. In all of the following cases, glucose is the only sugar. But in the case of inulin, students, we have a special type of carbohydrate called as fructose. Fructose is the main sugar. In the case of inulin. Clear, all of you? Any doubt there? Tell me, ask me quickly in the chat. Any doubt there? And heteropolysaccharide, we have peptidoglycan, NAG and NAM, and agar, which is used in your microbiology experiments. Biomolecules is getting over very easily, students. Now, where will the question come from? The main question always arises from the function. For example, starch. I told you already, it is a polymer of glucose. Yes. Food reserve in the case of plants, I mentioned starch in the case of plants, consisting of amylopectin and amylose. This is very important. This is very important. Right? Diksha sir, that joke never gets over, no? Diksha sir joke. Okay, so it is made up of amylopectin and amylose. So students, all of you know the structure, right? All of you know the structure of amylopectin and amylose. You will study that in your chemistry. That is alpha 1 linkage. So you have long chain of glucose. There is alpha 1614 linkage. Alpha 14 linkage in amylose, and amylopectin you have alpha 16 linkage. One down will come. You learn that in biomolecules in class 12th. Not now. Wait. Okay. Now glycogen, polymer of glucose. Again, see, glucose, food reserve in the case of animals. Compared to starch, glycogen is highly branched. Meaning of that is what? Meaning of that is more and more alpha 16 linkage can be found in the case of your glycogen. Chemistry also I'm teaching you now. What do you need? Huh? What do you need more? Chemistry also you're learning today. Okay. Now, stored in liver and muscles. Now, inulin students, inulin. Polymer of fructose, remember that. It's a PYQ. Right? Polymer of fructose is a PYQ. Asked you, like, you know, it is 3-4 times they've asked you. Okay. It is a polymer of fructose, glucose. Okay. It is present in the case of plants. Okay. Now, cellulose. Cellulose is again polymer of glucose, forms structural part of the plant. All of you know this. Now students, do you find cellulose in the case of plants? True or false? Tell me. Do you find this cellulose in the case of plants? Obviously, sir. What are you asking? Cellulose is found in the case of plants. Okay. Now chitin, where do you get chitin, students? It's a polymer of N-acetyl-glucosamine. It is found in the exoskeleton of arthropods. Looks so nice, no? Hari's favorite friend this is. Hari's favorite friend are blue beetle here. There is a movie on this. Go check it out. There is a movie called as Blue Beetle made on this beetle here, right here. Okay. What is the special power? The special power is exoskeleton of arthropod has chitin. That is why it is very strong. Very strong it is. Nice, very strong. When you add a nuclear missile on it, or you add also, nothing will happen to this guy. Okay. That is chitin. Okay. Now peptidoglycan we learnt. Bacterial cell wall, yes. N-acetyl-glucosamine, N-acetyl-muramic acid. Heteropolysaccharide. Okay. We learned agar-agar. Oh, looks like a nice water droplet, no? Agar-agar. This is the two different components, two different types of sugars of agar-agar. They will not ask you this in examination. Just telling you. But learn agar-agar is a heteropolysaccharide. Your peptidoglycan is a heteropolysaccharide. If I ask you students, if I ask you a question, chitin is a homo or heteropolysaccharide? Tell me the answer. Quickly. Chitin is a homopolysaccharide or a heteropolysaccharide? What would be the answer? It is a homopolysaccharide. Remember that point. That also is a NEET PYQ. Okay, NEET PYQ. Right? Chitin is a homopolysaccharide. Cellulose is a homopolysaccharide. Your glycogen, starch and inulin is also a homopolysaccharide. Done. Now, students, you might be wondering, sir, what is this reducing sugar? What is the meaning of a reducing sugar? How many of you have wondered this? Tell me. How many of you have wondered this? In chemistry, they have told you. In chemistry class, they have told you what is a reducing sugar. Understand? Answer is on the board. I know. I saw. That's why I asked you. Sometimes even the answer is on the board, you will tell the wrong answer. What to do? This is life. Okay. Clear? Now, we will start something called as reducing sugar now. Understand this concept is very simple. Very simple concept. Reducing sugar, okay? How many of you like free things? If someone gives you a free chocolate, you'll take it. Don't take it from strangers, obviously, but if your friend is giving you, take it. Okay, free chocolate, you love it. Right? Your Benedict solution also loves free things. So your aldehyde, sometimes what happens is, this aldehydes and ketones sometimes have your carbohydrates, some of the carbohydrates can have a free aldehyde and a free ketone. They can give away their aldehyde and ketone part. So your reducing sugar, students, your reducing sugar, mostly monosaccharides, have a free aldehyde and a free ketone group with which a reaction can take place. That is a reducing sugar. That's all. So a particular aldehyde, a particular carbohydrate, glucose, fructose, galactose, they have something called as a free aldehyde groups or a free ketone group. That is called a reducing sugar. Okay, all of understood? That is called as a reducing sugar. Reducing sugar has both free aldehyde or a ketone group. Now I'll tell you a simple experiment. You might have done it in your classes in chemistry lab. So if you take one test tube, in the test tube you need to add 2 ml of a glucose solution. All of you know that, right? Glucon D, one box you get glucon D powder. Don't take orange flavor, take a normal flavor, non-flavor one. Okay, take glucose powder, add 2 ml solution here. To this 2 ml solution you need to add equal amount of Benedict solution, equal amount. If I add 2 ml here, how many ml of Benedict solution, tell me? If I add 2 ml here, 2 ml of glucose solution, so I will add 2 ml of Benedict solution. Done, done, done. Now what we'll do, we'll keep it in water bath. We'll keep it in water bath. After some time, the Benedict solution, which is blue color, will turn into red color. Turn into red color. Why? The principle behind is very simple. That is, reducing sugar reduces the cupric ions. The cupric ions in the Benedict solution is reduced to cuprous ions. Cupric to cuprous ions. And it shows that the presence of reducing sugar. Presence of reducing sugar. Okay? Presence of reducing sugar. I'm live, no? For a second I thought the live stopped. Full scam I became. Okay? Done. This is called as reducing sugar. Now students, what if I add a polysaccharide? Now, what if I add a polysaccharide or a disaccharide? What will be the color change? Tell me now. Tell me. If I add a polysaccharide and if I add a disaccharide like sucrose, for example sucrose, will there be a color change? Tell me. Will there be a color change? Tell me, all of you. The stream is not stopped. No, I can see the chat, that's why. For a second I thought the stream stopped. I'm talking to myself from past 10 minutes I thought. Okay, tell me what will happen now. Tell me what happened. Tell me. Chemistry, someday. Will it become red or blue? Simple question. Answer. All of you would answer. Don't be hesitant. Don't be shy in my class. Tell me. If I take a small beaker, if I take a small test tube, I add polysaccharide and I will add Benedict solution to it. What will be the color change? Will it remain blue or will it become red? It will remain as blue color only. Why? Because students, if you look at the structure of your polysaccharides, there is no free aldehyde or a ketone group to react. You can see here, in this polysaccharides or a disaccharides like sucrose, there is no free aldehyde or a ketone group. So I will call polysaccharides and disaccharides as non-reducing sugar. Is that point clear to all of you? Understood? Non-reducing sugar. No free aldehyde or ketone group. No aldehyde. They have something called as reducing end, non-reducing end also. Remember that. Polysaccharide always has a reducing end, non-reducing end. Now, I will tell you one more experiment now. Ready? One more experiment. Ready? All of you tell me in the chat. Everyone ready for one more experiment? The second experiment is also very simple. Very simple experiment. Okay, very simple experiment. You take iodine solution. You would add the iodine solution to the starch containing sample. And you notice that the iodine solution, which was almost like a tincture, like a light blue, light yellow color tincture, it is becoming into blue color. Magic. Color changing is happening. The iodine solution, which was almost a light yellowish color, no, it's turned into blue color in the presence of starch. At the same time, if you take iodine solution and add it into a solution containing cellulose, there was no color change almost. There was no color change and it remained as yellow. What is the magic here? What is the trick behind the magic? The trick is very simple. The trick here is to look at the structure of cellulose and starch. Now, if you look at the structure of a starch, do you see some branching here, students? Do you see some branching? Tell me. Do you see some branching? Yes, obviously. Now, I'll ask you a simple question. Okay. I will throw this pen at you. I'll throw this pen at you and I will tie your hands. Can you catch it? Tell me. I'll throw this pen at you and I'll ask you to catch it. Will you be able to catch it if your hands are tied? Don't tell me you'll catch it with your mouth and all. Don't do all that. But will you be able to catch it with your hands? Obviously not. Your hands are very tied. Now I will leave your hands free. And I'll throw this pen again at you and ask you to catch it. Some of you will still not catch it. That is a different story. But some of you will be able to catch it now. Yes, some of you will catch it. Okay. So imagine this starch has nice arms. Can you see? It has nice arms. Your starch. If I throw the iodine at starch, will the iodine catch now? Tell me. Tell me, all of you. If I throw the iodine solution at starch, will the starch be able to catch the iodine in its large branches of tentacles? Obviously it will catch. Obviously it will catch. So when this iodine is caught by your starch, it will change the color into blue color. Change the color into blue color. While look at the structure of your cellulose. One arm distance. It looks like it is standing in 1 arm distance, no? It looks like it is standing in 1 arm distance. See, 1 arm is taken standing. Line by line they are standing. Now, can you see any branching here? Can you see any branching here, students? No. Hands are tied. Even if I throw the iodine at your cellulose, it will not be able to catch. Poor fellow, it will not be able to catch. That is why there is no color change here. That is, your cellulose cannot hold iodine. It cannot hold iodine, but your starch can hold iodine. Right? That is, starch can hold I2 and that's why positive blue color. While your cellulose cannot hold iodine, that's why negative test brown to yellow. Clear, all of you? Clear, all of you? Tell me. Is that basic chemistry? See, chemistry is so easy also. Sometimes chemistry is easy, but other times, if I teach organic chemistry, all of you will go mental. That is a different story, but yeah. All of you understood the concept now? Yes, got it, all of you? Now, I told you a small story, right? Can they ask it in examination? Can they ask this simple concept in examination? Can they ask this question in examination, students? The simple concept can be asked in examination. You might be wondering. Here is the question, students. This question right here is a NEET PYQ. NEET PYQ. Try solving, all of you. Tell me the answer in the chat, all of you. Tell me. Tell me the answer in the chat, all of you now. Question is very simple. NEET PYQ. Cellulose does not form blue color with iodine. All of you know cellulose hands are tied. Okay, hands are tied. It cannot hold iodine. Why? It breaks down when iodine reacts with it. It is a disaccharide. It is helical structure. It does not contain complex helicals. That is, it does not have branching, hence cannot hold iodine molecule. What is the answer now? What is the answer? Obviously 4. So you notice, students, you notice that even a simple concept can be asked as a question in this chapter. Okay? It's a NEET PYQ, all of you. NEET PYQ. Any doubt there, students? Any doubt there? If there is no doubt, can we read NCERT lines now, students? Iodine was done, right? Iodine is done. Can we read NCERT lines now? Can we start? So before we read polysaccharides, can we read now? Now all of you read with me now. All of you should read with me. Read loudly. Read loudly. And D for Diksha ma'am. Yes, in the chat. Students, I have heard that how many of you know Vedantu Telugu channel? You know, right? NEET Vedantu Telugu channel, right? Ajay sir is my friend, and we were speaking a couple of days back. Ajay sir and we were sitting in the pod and we were talking, have a friendly discussion and all. And he stays near my house also, Ajay sir. Okay. And he told me in his chat, all of you were telling A for amazing Gopika ma'am, B for Baswa sir. And I was shocked. I'm like, our students are going there. Our students are going there. There also you're telling our names. and sir was telling me, see sir, your students love you so much and they're telling your names in my chat also, right? So that made me really happy, and I told it Gopika ma'am. Gopika ma'am also was really happy, right? So when our students go and tell our name there, so yeah, it made us happy, all of us. Yeah. So now let's see the NCERT, all of you. Let's see the NCERT. And same thing even Shimon sir mentioned once. I was talking to Shimon sir, and even though our names were not mentioned, Shimon sir was telling me like how all of you are, some of our students who even watch his lectures there in Shimon sir. So it shows that, you know, right, it shows that our students are going in the Tamil channel also, we are going in Telugu channel also, right, and we're coming back here also. Coming back here also, right. Now let's start with polysaccharides now. Let's start with polysaccharides, right. Now tell me students, the acid insoluble pellet, tell me, acid insoluble, will it be present on the bottom or on the top? All of you know the concept, right? All of you know the concept. Tell me, acid insoluble pellet will be present on the bottom or on the top? Obviously on the top, acid insoluble. Yes, acid insoluble pellet also has polysaccharide. So students, you tell me right now in the chat, all of you, if acid insoluble is present on the top and we find polysaccharides on the top, what should be the molecular weight? Less than 1000 or more than 1000? Tell me, less than 1000 or more than 1000? Obviously it should be more than 1000. More than 1000. Can I call it retentate or filtrate? I will call it retentate because it is on the top. Yes, retentate on the top. Acid insoluble fraction. So can I call it biomicromolecule or should I call it biomacromolecule? In the chat, all of you. So I should be calling it biomacromolecule. Yes, macromolecule. So another cause of macromolecules, polysaccharides are long chain of sugars. We learned that. They are threads, literally a cotton thread. Cotton thread, like long cotton thread. Yes. Containing different monosaccharides as a building blocks. So what is happening here is, it is a long thread which has multiple monosaccharides joined together with the help of glycosidic bond. Yes, long thread. Yes, for example, cellulose is a polymeric polysaccharide containing only one type of monosaccharide. See, so cellulose is a homopolysaccharide. See, one type of monosaccharide, that is glucose. Cellulose is a homopolysaccharide. All of you know this. Starch is a variant of this but present as a storage. See students, did we write all this? Did we write all this storage and structure? See, it is mentioned here in NCERT, but my students, we learned it with the help of a proper difference like this. Proper difference. Can you see? Structure cellulose and chitin, but storage starch. Inulin, storage fructose. That's how you learn it. Okay, that's how you learn it. Now, starch is a variant of this but present as a storage house of energy in plants, tissues. Animals have another variant called as glycogen. Inulin is a polymer of fructose. Polymer of fructose in a polysaccharide chain. Say glycogen. Okay. In a glycogen, the right end is called as the reducing end. So always remember, right end is always reducing end. Okay, right end is always reducing end. So write down here, right end is always right for reducing, RR. The RR movie is there, no? Right end is the reducing end. So what should be the left end? Left end of a polysaccharide. See, this is the right end. This is the left end. Left end is called as non-reducing end. Non-reducing end. Non-reducing end. Clear? All of you? Clear? Clear all of you? So right end is always reducing end, left end is non-reducing end. Easy. Easy, right? Easy. Now, in a polysaccharide chain, say glycogen, the right end is called as the reducing end, the left end is called as non-reducing end. Okay. It has branches as shown in the form of cartoon. I will show you the cartoon now. Starch forms helical structure. See students, this is what we learned. Starch forms helical secondary structures. In fact, starch can hold I2 molecules in the helical portion. The starch I2 is blue in color. Cellulose does not contain. See, cellulose cannot hold it. The cellulose cannot hold it. cannot contain the complex helicals and hence cannot hold I2. So cellulose cannot hold I2, starch can hold I2. Starch can hold it. Is this part clear to all of you? Tell me yes or no in the chat. Sir, are you able to understand Telugu? Unfortunately, if you speak, I can tell, but reading a little difficult. Right? Okay? Is this paragraph clear to all of you students? If it is clear, I want everyone to add green hearts in the chat. If this part is clear, any doubt, tell me, ask me right now. No doubt? No doubt, all of you? No doubt? Yes. This is right here is the polysaccharide part. This is the cartoon they're telling me. I don't know what cartoon is this. I don't know. Tell me, is this cartoon? No? Tell me, is this cartoon? I don't think it's cartoon anywhere, but yeah. This is the representation of glycogen. Now, what I tell you here is glycogen is showing more branching here. Can you see? More branching. Alpha 1,6 branching can be seen here. Branching. This structure, this is amylose, amylopectin. This is the branching. Homopolysaccharide. Homopolysaccharide. Easy, easy, all of you. Now, plant cell wall is made up of cellulose. Okay. Paper made from plant pulp and cotton fiber is cellulosic. Therefore, more complex polysaccharides in nature. See, this is also PYQ. There are more complex polysaccharides in nature. They are building blocks, that is amino glucose and chemically modified sugar. Glucosamine, N-acetyl, galactosamine, and exoskeleton of arthropods, for example, a complex polysaccharide called as chitin. Students, remember examples. Examples. Complex polysaccharide. Complex polysaccharide. Complex polysaccharide. Chitin. Chitin. Then we have your glucosamine. 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 Then we also have the N-acetyl glucosamine. NAG. Complex polysaccharides. Complex polysaccharides. Clear? Clear? Okay. Sir, mine is lovely state Karnataka. Oh, all of you. So tell me students, how many of you are from Karnataka? See, I can understand Kannada really well. All of you know that, right? Obviously, I am from Karnataka only. If I don't know Kannada, then that will be a different story. But yeah, I can understand Telugu, Tamil and Kannada and Malayalam to some extent. Kannada I can speak very well. Telugu not very well speak, but yeah, I can understand. Because obviously, who doesn't watch Bahubali? Who doesn't watch RRR, right? We obviously watch... see, this is what I love about South India, right? Even though if I'm from Karnataka, I will end up watching Telugu movies, Tamil movies, Malayalam movies all the time. Even I watch Kannada movies. So when you watch such movies, we become bilingual. Multilingual, sorry. We become multilingual. That is the speciality of South India. That's what I love about us. We end up learning so many different languages at the same time. Right? All the time it happens. I'm not even kidding. It happens in the class also. When I was studying also, there were many students from different state. Kendriya Vidyalaya. Right? So we end up learning so many different languages. Right? Right. Prabhas fun, it seems. Okay. Now, with that being said, students, can anyone tell me the answer for this question? Anyone tell me the answer to this question? All of you. Let's see who tells the answer first now. Race is started. Yes, race is started. Protein will show which type of bond. Peptide bond. I'll give you a small clue. Peptide bond. Okay. Unsaturated fatty acids. Students, I'll give you one more clue. It is double bond. CC double bond. Okay. Unsaturated fatty acids. Saturated means a single bond. Saturated means single bond. Unsaturated double bond. Okay. Protein is going to be here in peptide bond. Now tell me. Polysaccharide. I told you. Polysaccharide will show polysaccharide will show glycosidic bond. Yes. A is 4, B is 1, and D is going to be 3, and nucleic acid is going to be phosphodiester bond. Who was the first one? I didn't understand. Madhumita was the first one, right? Yes. Anyone? Who told the answer first? I forgot. I couldn't see the chat. Chat went very fast. Boom, it went off. I couldn't see. Me first. Congratulations. So this is very easy question. It's option number 3. Easy, right? Now, third option. Now let's see who answers this question first now. With this answer, right, green hearts in the green heart also add with the answer. So I will know who answered first. All of you quickly. And some of you are wondering if I cannot talk Hindi. I can talk Hindi. I can, but it is a little thoda poda, turka murka it is. Okay, let's see who answers this question first. Inulin is a polymer of. Inulin is a polymer of. Parvati is saying for answer, huh? Ravindrani, exactly. Option, no, Madhumita was the first person to answer this. Fructose is a polymer of, inulin is a polymer of fructose. That is, a long chain of fructose is there. That is making up the inulin. That is making the inulin. Now, can anyone tell me the polymer of, can anyone tell me the structure of an amino acid quickly in the chat? Structure of amino acid. Does the amino acid has a carboxylic group attached to it? Yes or no? Quickly tell me in the chat. Amino acid, the carboxylic group. Does it have a carboxylic group? Yes. Does it have a amino group? Yes. Does it have a functional group? Yes, it has a functional group. Students, this is the last question now I'll ask you. If the functional group is CH2, CH3, what is the name of amino acid? Go. What is the name of amino acid? Go. No. If after you answer that question, next question is here on the board. Read the question, all of you. Yes, quickly in the chat, answer. I want all the answers. Is it serine or alanine? Is it serine or alanine? What is the answer? What is the answer? Is it serine or alanine? Think again and answer, all of you. Don't copy after one. Just because the first person is telling serine, all of you will copy serine. What is the answer? Tell me this answer now. Exoskeleton of arthropods is composed of. The blue beetle, all of you remember blue beetle. Blue beetle. It is chitin, exactly. Who was the first person to answer? Who was the first person to answer this question? Who was the first person? I forgot. This is a problem. The chat goes fast. Rohit. Rohit was the first person. Or Chotu? I forgot. I couldn't see. Okay. This one I'll see properly. Add red hearts with it. I will be able to tell. Add red hearts. I'll be able to tell. Okay. Which of the following is not a complex polysaccharide? Not a complex polysaccharide. Let's see who answers this question first. Let's see. Harshita. Okay. Who answers first? Not a complex polysaccharide. Chitin, glucosamine. Which is not a complex polysaccharide? Abhinaya. Option number 1. Exactly. Exactly. Abhinaya. Students, did I tell you here? Did I mention this somewhere? I think I mentioned somewhere, no? Did we mention here something, students? All of you said 4 option. One, do some 10 sit-ups. 10 sit-ups in the class, all of you. Who did told answer wrong? Complex polysaccharide, did I not mention here? Did I not mention here? Chitin, glucosamine and NAG. Chitin, glucosamine and NAG. N-acetyl glucosamine. Okay. So 3 I told you is a complex polysaccharide. 3 are complex polysaccharide. What is the question now? Tell me. The question is asking me which is not a complex polysaccharide. Not a complex polysaccharide. Tell me what is the answer now. Don't be shy. All of you who made the answer wrong, sit-ups in the class only first. What is the answer now? Tell me. Chitin is a complex. Glucosamine is complex. N-acetyl glucosamine is also complex. Yes. So what is the answer? This right here is a NEET 2024 question in which 50% of the students went wrong. And that is why I told you the answer also in my class. So what is the answer? 1 is the answer. Okay. All the students who made it right, I want everyone to clap for them. All of you clap for them who made it right answer. That is very important. Appreciation is very important. Inulin. Inulin is not a complex polysaccharide. Inulin is a simple polysaccharide, storage, polymer of fructose. Now, let's see who answers this question first now. Which of the following is most abundant protein in the animal kingdom? Abundant protein in the animals. I didn't teach you this, but you should know this. Most abundant protein in the entire animal kingdom. Entire animal kingdom. Most abundant in protein. Inulin, no, inulin is over. Most abundant protein in the animal kingdom. I think Madhumita was the first person to answer. It is going to be collagen. It is going to be collagen. It will come. I'll show you where it will come. Okay, I'll show you that. It is collagen is the answer right now. Collagen is the answer right now. I will show you where. Okay. So before we go that, students, can we read nucleic acids? Can we read about nucleic acids a little bit? We have already finished this. So we are just revising right now. We are just revising. Okay. Revising right now. Okay. Revising. The other type of macromolecule is that one would find in the acid insoluble fraction of living tissue is the nucleic acid. These are polynucleotide. Together with polysaccharide and polypeptide, these comprises the true macromolecules. So they'll ask you a question. AR question they'll ask you. AR question. Assertion is very simple. Assertion is going to be polysaccharides and polypeptides are the true macromolecules. They are the true macromolecules. Is the assertion correct or wrong? Tell me. Is the assertion correct or wrong? Tell me. All of you. See, a simple line of NCERT can be made into an assertion and you might be wondering where it asked from, right? The simple assertion is going to be polysaccharides and polypeptides are true macromolecules. Assertion is true. Yes, assertion is true. See, macromolecules of living tissue. The building blocks of nucleic acids are the building blocks of nucleotides. That also can be asked as a question, right? Nucleic acids are the building blocks of nucleotide. So when many nucleic acids join together, they make the nucleotide. A nucleotide has three chemical distinct components. One heterocyclic component, that is the sugar, and the second is the monosaccharide, and the third one is a phosphate. All of you know, right? A nucleotide is made up of a sugar, phosphate, and a nitrogenous base. A sugar, a nitrogenous base, and a phosphate group. Nucleotide. Now tell me, students, in nucleotide and nucleoside, what is that one missing thing? I've told in the last class. In the last class I mentioned between nucleotide and nucleoside there is one major difference. What is the missing thing? All of you. What is the missing thing between nucleotide and nucleoside? That one missing link. Well, keep reading, I'll tell you. I've told you in the previous class, let's see. The heterocyclic compound in nucleic acids are nitrogenous bases named adenine, guanine, uracil, and cytosine, thymine. Adenine and guanine are substituted purines. Adenine and guanine are purines. Rest are pyrimidines. Cut pyramid. Remember cut. CUT. Cytosine, uracil, thymine are your pyrimidines. Exactly. Phosphate group is missing in your nucleoside. Phosphate is missing. Okay. The skeleton heterocyclic rings are called as purines and pyrimidines respectively. The sugar found in the polynucleotide is either ribose sugar or a deoxyribose sugar. Now tell me the difference now. What is the difference between a ribose and deoxyribose? In the last class I told you. In deoxyribose, one oxygen is missing. For example, students, I think I have the structure also here. If you look at the structure of deoxyribose, one oxygen is missing in the second carbon, deoxyribose. And deoxyribose is present in the case of your DNA. Deoxyribonucleic acid. And ribose is present in the case of your RNA, in which in the second carbon, one oxygen is being present here. Oxygen is missing in the case of your deoxyribose. Difference is very clear. Difference is very clear for all of you. Yes. It's called as DNA and RNA. DNA and RNA. See, purines are, and remember students, purines are double ring structures. All of you remember? Purines are double ring structures. Double ring. Guanine and adenine both are double ring structures. That also is a question. Famous question. Famous question. Your pyrimidines are single ring, cytosine, thymine and uracil. That is cut. Cytosine, uracil, thymine. Cut the pyramid. Cut the pyramid. Pyrimidines. Okay. Now, let's see the proteins now. Any doubt there, students? Any doubt there? Any doubt in nucleic acid? We have finished nucleic acid in the previous class, that's why I went a little fast there. Okay, any doubt there? Quickly ask me. Any doubt there? So with that being said, let's read about proteins now. Proteins are polypeptide. Okay. Now what is the meaning of polypeptide? Students, tell me. Are proteins now micromolecules or macromolecules? Are they micro or macro? Tell me. Are they micro or macro then? Or is protein made up of a single peptide or many peptide? Tell me. Is a protein made up of a single peptide or many peptide? Simple question, no, simple question. Is protein made up of a single peptide or many peptides? And those of you watching my session for the first time, in my class I always ask many questions. Okay, tell me. So it is made up of many peptide. That is why it is called as polypeptide. Can you see? Polypeptide. They are linked, they are chains, linear chains of amino acids linked by peptide bond. Okay. Each protein is a polymer of amino acid. There are 20 different types of amino acids. Now tell me, students, all of you remind me now quickly. Out of this 20 amino acids, how many are neutral, how many are acidic, and how many are basic in nature? How many are basic in nature? Quickly in the chat, all of you. Out of this 20 amino acids, 15 are going to be neutral, 3 are going to be basic, 2 are going to be acidic in nature. Acidic in nature. Glutamic acid, one is glutamic acid, other one is aspartic acid. Easy. Okay. Adenine, cytosine, proline, tryptophan, lysine, etc. A protein is a heteropolymer. Very important line. Protein is a heteropolymer. Protein is a heteropolymer. Very important line. Heteropolymer. Okay. It is a heteropolymer. Protein is a heteropolymer and not a homopolymer. A homopolymer has only one type of monomer and n number of times. This information about amino acid content is important, as later you will in your nutrition lesson you will learn that certain amino acids are essential for health and they have been supplied to our diet. Hence dietary proteins, whey proteins, are source of essential amino acids. Therefore amino acids can be classified into essential and non-essential. Essential should be taken from outside, non-essential are produced in our body. Important, easy. Okay, produced in the body. Clear? Yes. The latter, you know, which are produced, which our body can make while we get essential amino acids through our diet. Okay. Now, functions. Function is written here. Very important. This is the function. This right here is the function. Function of amino acids. Functions. Understand the functions. Okay. Protein carry out many functions in the living organisms. Some transport nutrient across the cell membrane, some fight, that is your GLUT4, some fight infections, antibodies. Some are hormones, different hormones in your body are basically type of proteins. Some are enzymes, See, collagen is a ground substance, trypsin is an enzyme, insulin is a hormone, antibody is the infection type, receptors are many are there. GLUT4 is a transporter. GLUT4 helps in transporting different chemicals into the cell. GLUT4. This table, quickly read it in your mind. I'll give you 10 seconds. Go through this. 10 seconds, all of you. 10 seconds, go through it quickly, all of you. One nice fight is going on in the chat, I can see. One nice fight is going on. Keep fighting. In the exam day also, you'll keep on fighting like this only. Huh? Nice fight is going on in the chat, I can see. Now tell me, what is all of it done? Done? Collagen, enzyme, hormone, receptor, GLUT4. Done? Yes. Now here I'll tell you a few things here. Understand. The first thing which you need to notice here, students, is collagen is the most abundant protein in the animal world. So the most abundant protein a more abundant protein in animal world. In animal world is going to be collagen. It is going to be collagen. Now the next thing is going to be the most abundant protein in the entire biosphere. Most abundant protein in the entire biosphere, right? Most abundant protein in the biosphere is going to be Rubisco. Enzyme. Rubulose 1,5 bisphosphate carboxylase oxygenase. Now I'll ask one question now. Okay. Tell me, Rubisco, where do you find Rubisco, in which cycle? C2 cycle, C3 cycle, C3 and C4 cycle, or in all of them? Tell me, where do you find Rubisco enzyme? In which cycle do you find? Very good question this is. In C2 cycle, C3 cycle, C4 cycle, or all of them? Tell me, where do you find Rubisco enzyme? Quickly go chat. All of you tell me in the chat. Where do you find Rubisco enzyme? Students, do you not remember? Yes, in C3 cycle, first stage we have Rubisco. Will the Rubisco bind with oxygen and produce PG and PGA in C2 cycle also? Yes, in C2 cycle also. Do you not remember in your C4 cycle, technically even C3 cycle is also happening? Yes, in C4 cycle, in the case of your bundle sheath cells, C3 cycle is also happening. So if C3 cycle is also happening, Rubisco is also present there. So can I tell? Rubisco is present in C3, C2, C4, all the cycles. Tell me your answer now. Where do you find Rubisco? See, such a good question. This question came from nowhere. Suddenly, right? So it can be found everywhere. Okay, Rubisco. Now tell me, when CO2 and oxygen is present in equal level, Rubisco will bind to CO2 or oxygen. Where will Rubisco bind? Tell me, where will Rubisco bind? Tell me, where will Rubisco bind when oxygen and CO2 are present in same level? Obviously, it will bind to CO2. Right? Now, what will happen if the oxygen level goes up? Then where will Rubisco bind? If oxygen level goes up, your Rubisco will bind where? Tell me. Rubisco will bind to oxygen that time. So go watch that lecture. There is something called as when will C2 cycle occur? That is very important. Go watch that lecture if you do not know. Okay? Please watch that also. Back to a question. Time starts now. Let's see who answered this question first. Time starts now. Quickly, go in the chat. Who answered this question first? It's a NEET 2024 question. NEET 2024 question on your board right now. Time starts. Go. GLUT4. We just now learned GLUT4 helps in transportation. So A is going to be 4. Yes. Insulin is a hormone. We just now learned. Trypsin is an enzyme. Collagen is a intercellular ground substance. D. Exactly. D or 4 rubber option. Yes. Absolutely. So we learned now GLUT4 is going to be a transportation. So when I say A is going to be 4, so there's only one option left, right, Paul? There is going to be only one option left. So option number 4 is right answer. Yes, Sonu, Soni, Rohit. Yes, Rohit is the first person. Lightning mode. Understood? So if you just knew the one option, you could be able to answer this question. Now students, now that is the end of polysaccharides, that is the end of nucleic acid, that is the end of carbohydrates. Now can we start with the structure of protein? Tell me in the chat. Can we start now? Structure of protein now. Full fight is going on. Me first, it's fine. Okay. Can we start now with protein now? Structure of protein. Till now the class is going very smooth. All of you are answering my questions. We are learning something new every single step. Now we will learn something called as the structure of protein now. Ready? Okay. Can we start? Can we start now? Structure of protein now? Yes. Now students, structure of protein is not very simple actually. A protein has multiple structure. A protein has multiple structures. There is something called as a primary structure, secondary structure, tertiary structure, and a quaternary structure. And each structure has an importance. For example, students, now you tell me, look at the primary structure. All of you look at the primary structure. Focus, ultimate focus here. Tell me, is the primary structure linear, or is it branched, or is it showing any type of bending? Tell me. Look at the primary structure, all of you. Tell me. Look at the primary structure and tell me, is it linear or is there any change, is there any branching here? You tell me, sir, linear, primary structure is linear in shape. It's linear. It's linear in shape. Primary structure is linear in shape. Okay, agree. Now, in primary structure, what do you understand from primary structure? What do you understand from primary structure? Listen to me very carefully. When all of you are standing in one line, now imagine we take Gracie, we take Rohit, we take Ravindrani, we take Chotu, we take Chitra, and we make all of them stand in one line. If I make all of you stand in one line, and if I look from very far, and all of you standing like this, horizontal, all of you standing in one line like this, and if I'm looking at all of you from one angle, can I tell exactly where Ravindrani is standing? Can I tell exactly where Gracie is standing? Can I tell exactly where Paul is standing? Can I tell exactly where your Pro is standing? Can I tell? Can I tell? Where exactly each one is standing? So your primary structure tells you the position of each amino acid. So on the linear chain, you can tell me this first, second, third, fourth, fifth, sixth, seventh, the position of each amino acid. The position of each amino acid can be understood with the help of the primary structure. Is that point clear to all of you? Tell me yes or no in the chat. Is that point clear to all of you? Yes? Point is clear to all of you? That is basically the primary structure, which tells you which amino acid is present where. The position of amino acid. Position of amino acid. Now, understand the secondary structure now. Now you take primary structure and you give it a shape. Now I will do a whistle, I'll make a whistle. All of you will twist and turn. All of you will twist and turn and you will make a secondary shape now. You will make a secondary shape. What is happening in secondary shape, students? All of you know basic physics. All of you remember solenoid. Solenoid in your physics, solenoid. The coiling structure, solenoid. This right here, alpha helix structure is like the solenoid structure. Can you see? Solenoid structure. Alpha helix. Alpha helix. So all the protein is attached in a alpha helix structure, and the other one is something called as beta pleated sheets. Beta pleated sheets. Now what is happening in beta pleated sheets? Students, understand. Can you see this? Can you see this, students? Are you able to see this? This right here is called as, this is the shape. This is the shape right here of the beta pleated sheets. Beta pleated sheets look like this. And from here, we only have amino acid here. I told you we have amino acid here and we have one amino acid here. How are they attached? They are attached with the help of peptide bond. Peptide bond. Yes, peptide bond is there. Now when we come to secondary structure, there is also hydrogen bond here. We have the hydrogen bond. Hydrogen bond here. Hydrogen bond. That is your secondary structure. That is your secondary structure. Now we come to the tertiary structure now. Now what you need to imagine, all of you close your eyes and think. Just close your eyes and think. You're taking one alpha, you're taking one beta and bringing them close and making a woolen ball. And imagine, you will take one alpha helix structure. You will take a beta plate structure and you'll bring them together and you'll make a nice ball. What you're trying to do is basically you're making a ball like this. You're making a woolen ball like this. You see? You're making a ball. This right here is the structure of a tertiary structure of a protein. Right here, like a ball. Right, like a ball. So, can you see here? We have alpha here and we have alpha helix. We have beta plate also here. Both are present here, like a woolen ball. Now, before, this was around 2D. This was 2D structure. 2D structure. While your tertiary structure, students, while your tertiary structure is going to be a much more like a 3D structure. It's going to be something like a 3D structure. 3D structure. And most of the enzymes, students, most of the enzymes are biologically active are your tertiary structures. Tertiary structures. Understood, all of you? Understood now? Tell me yes or no in the chat quickly. Understood? Now, understand quaternary structure now. Understand quaternary structure. Very simple. Now, this is a tertiary structure right here. This right here is a tertiary structure. I have one more tertiary structure. When I bring two different tertiary structures, I get the quaternary structure. One tertiary structure, one more tertiary structure. They're coming together and they're making the quaternary structure. Can you see here? There's one chain here. There's one chain and two chain. Both are coming together. That is the quaternary structure. Quaternary structure. Clear, all of you? Tell me. In tertiary, we also have disulfide linkages. Yes. Along with hydrogen linkage. Hydrogen linkage also is present in the case of your secondary also. Clear? Is that point clear to all of you? Tell me. I'll read it. I'll show you one more time. I'll clear the doubts one more time. See here. Primary structure is what? Linear chain represents the position of the amino acid. Position of amino acid. Now here we have something called as your N-terminal and C-terminal. N-terminal and C-terminal. Students, why is it called as C-terminal here? Students, can you tell me? C-terminal because we have the carboxylic group here. Carboxylic group. That's why it's called C-terminal. We also have the N-terminal. N-terminal. N-terminal will have the amino group. Amino group in the N-terminal. Okay? So C-terminal, free amine group. Then C-terminal, free carboxylic. N-terminal, free amine group. Free amine group. Clear? All of you clear with that point? Three different points about primary. Now what about secondary? Folding of polypeptide chain due to interaction between the amino acid. So amino acids are interacting. There is hydrogen bond. So we have the alpha helix. We have the alpha helix and we also have the beta plated sheet. Beta plated sheet. Then we have tertiary structure. That is three-dimensional, protein fold upon itself, biologically active. All the biologically active here. Quaternary structure, what happens? We have one more tertiary. Joined together, we have the quaternary. That is, two or more polypeptide chains are coming together. See, arrangement of each folded polypeptide chain with respect to each other. One example is hemoglobin. Hemoglobin is an example of what? Hemoglobin is an example of quaternary structure. Can you see here? Here we have four chains. First chain, second chain, third chain and fourth chain. There are four different chains here. We have two alpha chains and two beta chains. Four chains are coming together and they're making the quaternary structure. Quaternary structure. Clear? If that is not clear, I'll make you read NCERT again. Read NCERT with me now. It'll become even more clear to you now. Read. Proteins are mentioned as earlier as heteropolysaccharide containing strings of amino acid. The structure of molecules means different things in different contexts. For example, Diksha sir, in inorganic chemistry, the structure invariably refers to the molecular formula in chemistry. NaCl, MgCl2, sodium chloride and magnesium chloride. The organic chemistry, what is meant? It is always written in two-dimensional view of the molecules. For example, we have benzene, naphthalene. Physics, Shreyas sir is coming here, Shreyas sir. Physics conjure upon the 3D views of molecular structure, while biologists, me and Gopika ma'am, biologists describe the protein structure at 4 levels. See, biology we are learning at 4 different levels. So biology is much more complex. 4 different levels, right? 4 different levels. The sequence of amino acid, the position of information of protein in which the first amino acid, which is and second, is called as the primary structure. So what is primary structure telling you? Primary structure is telling you the position of amino acid. See here, the sequence of amino acid, the positional information, which amino acid is present where in a protein, which is the first amino acid, which is the second, and so on. That is giving you the primary structure. Primary structure. Yes? Clear, all of you? Primary structure clear? Here. Primary structure is telling you the position of amino acid. Which amino acid is present where? Which is the first, which is the second, which is the third, which is the fourth. Primary structure. Primary structure. Okay. Now, of the protein. A protein is imagined as a line, linear protein. The left end is called as what? The left end is the first amino acid and the right end is the last amino acid. The first left end, the first amino acid is called as N group. So N is first. Remember, N is the first. The first amino acid is also called as the N-terminal amino acid, and the last amino acid, this is very important, this is a PYQ. PYQ, right here. And the last amino acid is called as the C-terminal because it has a carboxylic group. So remember, first is N always. First is the N group, N-terminal. N-terminal. Last is the C-terminal. C-terminal. Remember these points. Remember these points. Okay. A protein thread does not exist throughout as an extended rigid rod. A thread is folded in form of helical, similar to revolving staircase solenoid model. Okay. Of course, only some portion of the protein thread are arranged in the form of a helix. In proteins, only a right-hand helix are observed. Very important line again. You can only see a right-handed helix always. The other regions of protein thread are folded into a form of what is called as secondary structure, that is beta plated. Okay. The long protein chain is also folded upon itself into a hollow woolen ball, giving the tertiary structure. It is also 3D, I told you, 3-dimensional view of a protein. Tertiary structure is the absolutely necessary for many biologically active proteins. So if we have a biologically active protein, it should have which form? Tell me, all of you in the chat. If a biological protein should be active, it should have the tertiary structure or the 3D structure. 3D structure. It is compulsory. It is compulsory. It should have a 3D structure. Clear? This is a very important lines here. Very important lines. Very important lines. Very important lines. Okay. So tertiary structure is always compulsory for a biologically active protein, that is mostly the enzymes. Enzymes should have a tertiary structure. Tertiary structure. Students, do you realize every single line of NCERT I am telling you what is important, from where questions can come from? Questions can come from. Beautiful. Some proteins are assembled more than one polypeptide. I told you, one tertiary, second tertiary. Together they are called as quaternary. Quaternary. Okay. Quaternary. Quaternary. The manner in which these individuals fold polypeptide or subunits are arranged with respect to each other, linear string, spherical arranged upon itself, everything is the architecture of protein, otherwise called as quaternary. The adult hemoglobin consists of 4 chains. I told you, 2 alpha, 2 beta, 2 alpha, 2 beta, right? The 2 of these chains are identical to each other, hence 2 subunits of alpha and 2 subunits of beta together consist of your human hemoglobin. This can be asked as a question. This can be asked as a question in next year. I'm thinking prediction is going on already. Okay? Any doubt there? Any doubt there, friends? Tell me. Any doubt there? Okay. Done? Habba, I'm out of breath. I don't know why today. Out of breath already. So much of talking is happening. Any doubt there? Any doubt, the students quickly ask me in the chat. Block Chotu, why? What happened? What did Mr. Chotu do? Chotu Motu, what did he do? Students, every single line of NCERT has been covered till now. I believe there should be no doubt here, right? No doubt here. No. Any doubt till now? Students, tell me. Ask me. Any doubt? Emitting some water. No doubt? Okay, amazing. Done already, don't worry, it's already done. Already done. Yeah, that's already done, don't worry. You don't have to worry about it. So, now we are going in a very smooth direction. Right, we are now entering into enzyme part. Before we enter into enzyme part, there is a small paragraph which we will read now. Okay, all of you know this. In a polypeptide of protein, amino acids are linked by peptide bond. All of you know. Yes, peptide bond. We do a peptide bond, in which carboxylic group of one amino acid reacts with NH2. So COH plus NH2, water is being removed out. Okay. The next amino acid with the elimination of water moiety. Okay. It's what's called dehydration. Chemistry, you have learned this. In a polysaccharides, individual monosaccharides are linked by glycosidic bond. All of you know this. This bond is also formed by dehydration. This bond is formed between two carbon atoms of adjacent monosaccharides. So monosaccharides come together, we get the disaccharide. Hey, this question is repeated, no? Okay. All of you know this. This is collagen. So all of you read this question, and I'll just go out. I'll get some more water and come. Okay. I'll get some more water and come. Let's see who can answer this question. Friends, quickly smash that like button. What is this? No one is marking attendance, huh? No care only, huh? Quickly mark the attendance, all of you, and tell me the answer to this question now in the chat, all of you. A protein is imagined as a straight line. The left is represented by first amino acid, right is represented by last amino acid. Adult hemoglobin consists of 4 subunits. What is the answer now? Tell me. All of you discuss nicely, no? All of you discussed also nicely, I feel, the answer also. Okay. A protein is imagined as a straight line. The left end is represented by first amino acid. Okay. Did we learn that somewhere, students? Did I make you write down? First one is the N terminal. Last is the C terminal. First amino acid C terminal, so obviously wrong. Should be N terminal here. The right end is represented by last amino acid called as N terminal. No. Last is C terminal. Last is C terminal. So statement 1 is wrong, second one is true. 1 is the answer. A for amazing copy, come ma'am. Time starts now. Oh, this question is repeated. This question has been repeated. Oh. All of you can read the question, read the question. If you cannot, I'll read it for you. First Rohita, then you, okay? Okay. I will believe you. I will believe all of you. Okay. Which of the following bond is formed as a result of reaction of carboxylic group of one amino acid with amino group of other amino acid with elimination of water? Basically, which bond is present between the two amino acid? Which bond is present between two amino acids? Tell me. Which bond is present between two amino acids? Madhumati is saying option number 3 here. Can't read, huh? Sorry. I will send you the PPT and read that time. I will send you this presentation on my Telegram channel. All of you are aware, no? Quickly join, don't wait. You will, every single day you will get motivation quotes there. Every single day you will get nice information. I will always interact with you there. Any doubt you can always talk to me there. Right? The question is saying, which of the following bond is formed as a result of carboxylic group of one amino acid? Carboxylic group of one amino acid with amino group of other amino acid. Students, did we learn here? Amino acid linked by peptide bond, carboxylic acid group of one amino acid reacts with amino group of other amino acid with elimination of water. Peptide bond. Peptide bond. It's given right here. We learned it here only. Okay. How many of you are not in Telegram channel already? Quickly join Telegram channel. So what is the answer? Peptide bond. Peptide bond. 4. Peptide bond. Easy question. Easy question. Now students, with that, your all chunnu-munnu topics are over. All chunnu-munnu easy topics are over today. Now we will go to the difficult topics, the challenging topics, which are also easy, which are also chunnu-munnu. Okay, can we start now, students? Can we start with the little more interesting parts? That is, a protein has many functions. All of you learned. It has a structural function, contractile, nutritional, regulatory, defense, transport. We learned all this. We learned all this. But there is also one main function which we will be addressing right now. That is the catalytic function. That is going to be the catalytic function. So can we start now, students? The catalytic functions of a protein called as a catalyst. Called as a catalyst. Very easy concept. We will start very slowly, very sweetly. We will encounter each and every concept, kill every single concept, go to the next one. Okay. So can we start, students? Can we start with the catalyst part now? Can we have the catalyst part? Now students, tell me, what is a catalyst? What is a catalyst? Chemistry, all of you know chemistry. If you don't answer, I'll complain to Diksha ma'am. What is a catalyst? Catalyst is a chemical compound or a substance which can alter the rate of reaction. Tell me. Yes or no? It is a chemical substance. It is a chemical substance which can alter rate of reaction. It is a chemical substance. It is a chemical substance which can alter the rate of a reaction. Rate of a reaction, right? It can increase the rate of reaction or decrease the rate of reaction. Yes, increase or decrease the rate of reaction. Now, tell me, does it also get involved in the reaction? Does it get consumed in the reaction? No, it does not get consumed in the reaction. It does not get consumed in the reaction. That is, it can increase or decrease the rate of reaction without getting consumed. Without getting consumed. Okay. Now here we have two types of catalyst. One is called as inorganic catalyst. Other one is called as organic catalyst. The inorganic catalyst example is your platinum. Simple example is the platinum. Under organic catalyst, trypsin, which is basically a enzyme. So in this chapter, we'll be talking about the organic catalyst, that is an enzyme. That is going to be a enzyme. Now students, remember, I've told you this before also. What are enzymes? Are they protein? Are they lipid? Are they carbohydrate? Are they nucleic acid? What are enzymes? Tell me. Students, what are enzymes? What exactly are enzymes? So can I tell students, all enzymes are proteins, All enzymes are proteins except few. Except ribozyme. Except the ribozymes. Right, except the ribozymes. Why? Because ribozymes are basically RNA enzymes. Ribozymes are made up of RNA. They're RNA enzymes. Ribozymes are your RNA enzymes. So except ribozymes, all the enzymes are basically proteins. Proteins. Now tell me, what will happen if I heat the protein too much? The protein will denature. If I keep the protein in an ice cold condition, the protein will become stagnant. So similarly, if I heat the enzyme very much, will the enzyme even survive in the extreme hot conditions, or the enzyme will completely denature? The enzyme will completely denature because it is made up of protein. Right? It is made up of protein. Now students, some of you have a favorite hero, right? Tell me your favorite hero. Tell me in the chat. Favorite actor, sorry, not hero. Favorite actor. Tell me, who is your favorite actor? Now, all of you are favorite actor. If someone tells something bad about the actor, you will go all mad. You're like, how can you tell? How can you tell about Prabhas like that? You will tell no. You will tell, right? So similarly, how you have a favorite actor, your enzymes also have a very specific partner they'll choose. They are highly specific in action. They are very highly specific in action. They're very highly specific in action. Okay, specific in action, highly specific in action. They will never make sure. They will make sure that they'll only join to a particular substrate. One enzyme will fix to only one substrate. Very specific in action. Okay, very specific in action. And they mostly work in aqueous medium. They need a medium to work. They cannot work all the time. They need a specific medium to work. Okay, very specific medium to work. Now, something called as active site. Now, this is one small concept which everyone misunderstands or try to by heart this, but I'll teach you what exactly is a active site now. Okay. Now students, if you remember, when I was teaching you about tertiary structure of protein, in tertiary structure of protein, the protein is active, biologically active, it is 3D in nature. But did we also learn that a tertiary structure of protein will have certain pockets or crevices? Did we learn in tertiary structure of protein, a tertiary structure of protein will have small pockets? I didn't mention here. Okay. It was not given in NCERT also. Okay, it is not given. So let me tell you. So tertiary structure of protein, students. A tertiary structure of protein will have a small pockets or small gaps. For example, this is your, let's say this is a structure of protein. This is a structure of your enzyme. In this enzyme, there are this small gaps or pockets. Can you see? There are these small gaps or pockets in the enzyme. In the small gaps or pockets, there can be certain functional groups. There can be certain functional groups. Now these gaps, can you see these small gaps or crevices? These small gaps or crevices in the enzyme, in which your substrate will come and join. Substrate will come and join in this particular pocket. It will come and sit here. This particular pocket or crevices is called as the active site. It is called as the active site. Active site. Clear, all of you? Clear? Understood, all of you? Oh, you're telling real hero Baswa sir. Oh my god. Tell me, what do you want in Britain? Why so much of flattery? Huh? You want chocolate? Tell me. What do you want? So much of flattery is happening in the chat. So much of flattery is happening in the chat. Tell me, what do you want? Full butter you're putting. Huh? So this small gaps in the structure of an enzyme is called as an active site, in which the substrate will come and join. Okay, will come and join. Clear, all of you? Okay, so crevices or pockets in the structure of the binding site of the substrate, specific for substrate. It is very specific for substrate. Nothing else can come and join. Okay, only a substrate will be able to come and join there. Okay, catalyst reaction at high rate. Okay, that is an active site. Let's see what is given here. Almost all enzymes are proteins. There are some there are certain nucleic acids that behave like enzymes. They are called as ribozymes. So except ribozymes, everything is made up of protein. Okay. Now, one can depict an enzyme by line diagram. An enzyme can be any protein. See, an enzyme can be any protein, has a primary structure, that is amino acid sequence of protein. An enzyme can, like any protein, has a secondary structure and a tertiary structure. Point clear till now? So an enzyme can have a tertiary structure like a protein. Okay. When you look at the tertiary structure, is always given here. When you look at the tertiary structure, you will notice that backbone of the protein fold chain, backbone of the protein chain folds upon itself. The chain crisscross itself, and hence many crevices of pockets are made. So in the tertiary structure of protein, there are small crevices, means small gaps, and pockets are made. One such pocket is the active site. An active site of an enzyme is the crevice or a pocket into which substrate is fitting. Understood, all of you? It's given here only. I'm not making it up. In the tertiary structure of protein, there are small gaps or crevices. In that small gaps and crevices, a substrate will come and join. Is that point clear to all of you? Tell me. Is this clear to all of you? Thus, enzyme, through their active site, catalyze the reaction at high rates. Enzyme catalysis differ from inorganic catalysts in many ways. But one major difference needs to notice. That is, inorganic catalysts like platinum and all work efficiently at high temperature and high pressure. See, inorganic catalysts like Diksha is, imagine I am bringing one catalyst, Diksha ma'am is bringing one catalyst. Diksha ma'am's catalyst from chemistry background, it can work in high temperature and high pressure. But my catalyst which I am bringing, it is not able to function at high temperature. No. Why? Because the catalyst which I am bringing is made up of protein. And all of you know, protein cannot work in high temperature and pressure. So biocatalyst, the enzymes, cannot work at high temperature and high pressure. Is that point clear to all of you? Tell me. All of you remember that? Will you remember that? The biocatalysts, the enzymes, cannot work in high temperature and high pressure, except one. Except one. There is one enzyme which is acquired from a hot spring. from a hot spring, right? From a hot spring. The enzyme is acquired from a organism called as Thermus aquaticus. Thermus aquaticus. Thermus aquaticus. The enzyme name is dash. What is the name of enzyme? Rashid is saying all is well. So much of drama. Tell me, what is the name of enzyme? What happened to my catalyst? Denatured. But I have a Brahmastra with me. I have a special catalyst now, students. I have a special catalyst which can even work at high temperature, high pressure, acquired from hot springs. So Diksha ma'am defeated me, right? So I became very angry. So I went to a hot spring, put my hand inside, pulled out a new enzyme called as Taq polymerase. This Taq polymerase can work in high temperature and high pressure, and I show you to Diksha ma'am. See ma'am, look at this. Look at my enzyme now. Just like inorganic enzymes, inorganic catalyst, my enzyme can also work in high temperature and high pressure, called as Taq polymerase. See here. However, enzyme isolate from organism who normally live under high temperature, hot vents and sulfur springs are stable and retain their catalytic power even at high temperature. Thermal stability is thus an important quality of such enzymes isolate from thermophilic organisms called as Taq polymerase. My revenge is done here. Okay. So much of storytelling today. I don't know why. Now students, I told you, yes, enzymes can work really well. Enzymes can work really well, but how much are they efficient? Now imagine you're sitting for 1 hour. Tell me, in 3 hours, how many chapters can you finish? Tell me. In 3 hours, how many chapters can you finish? Tell me. Cooked stage, I know. It will become cooked, I know. In 3 hours, how many chapters will you finish? Tell me. In 3 hours, you can finish 1 chapter, 2 chapter, 3 chapters. 1 hour per chapter, you can finish somehow. Okay. 1 hour per chapter. Now, here we have one reaction. Here we have a simple reaction. CO2 plus H2O gives H2CO3. H2CO3. Okay. In this normal reaction, we are getting around 200 molecules in 1 hour. 200 molecules in 1 hour. Okay. Now imagine, how many of you watched the Chitti robot? How many of you know Chitti robot? Chitti. Now imagine you become like Chitti robot. Now you tell me, how many chapters can you finish in 3 hours? This enzyme is like Chitti robot. Okay. Now here we had 200 molecules in 1 hour with catalyst. Without catalyst, how much, students? 200 molecules in 1 hour without catalyst. Now with catalyst, what will happen? Like how Chitti robot is able to scan everything and finish. He can finish your entire NEET syllabus in what? 10 minutes or 1 minute. With catalyst we are making, with catalyst right here we are making 6 lakh molecules. 6 lakh molecules in 1 hour, 1 minute, 10 minutes, in 1 second. In 1 second we are able to make 6 lakh molecules. 6 lakh molecules. Here before, without catalyst, 200 molecules in 1 hour. All of you right now. But Chitti robot, how if Chitti robot is there, imagine even with enzyme, the same reaction with the enzymes, 6 lakh molecules in just 1 second. In just 1 second. 1 second. That is the power of a enzyme. Now you tell me, enzymes are powerful? No. Enzymes are powerful. Enzymes are incredible. Incredible power of enzyme. Incredible power of an enzyme. It can literally make 6 lakh molecules in just 1 second. 1 second. No. Yes, let's read what is given here. A physical change simply refers to change. Okay, this is all that is normal. Here they're telling you about something called rate of reaction. Rate of reaction, they'll never ask you all this. Rate of reaction. Okay. Now they'll tell you here is rate of reaction depends upon the temperature. Increase the temperature, rate increases. Decrease the temperature, rate decreases. That's all. Don't underestimate the power of enzymes. Exactly. See here. Rate can be called as, what is rate of a reaction, students? Rate of a physical or chemical process refers to the amount of product produced per unit time. In 1 hour, how much of product you are making, that is called a rate of a reaction. If you add direction to it, it becomes velocity. Physics, simple physics. Rate of a reaction. Okay. Every 10 degrees Celsius, it changes. It changes every 10 degrees Celsius. Increase by 10 degrees Celsius, 10 degrees Celsius, rate will increase by double, rate will become double. Decrease by 10 degrees Celsius, it'll become half. Rate will become half. Simple math. Okay, simple math. What I told you is right here. 200 molecules in 1 hour, 6 lakh molecules in every second, right here. It's given right here. Enzyme is rocking here now. Yes. And remember, students, enzymes, when the reaction is happening, in case of biology, if you remember glycolysis, it doesn't happen in one step. It happens in 10 different steps. Glycolysis, remember, 10 different steps were there in glycolysis. So it is a can you see it? Multi-step chemical reaction is happening. And every single step, there is a different type of enzyme. Okay. So please update physics PYQs, sums in English. Darshini, what, if you learn physics from me, you will only go mental. Huh? You learn physics from me. What days have come? Okay. Now, this is the same thing. We can skip this. You can read this on your own. They're basically talking about respiration in plants chapter here. Okay. Alcoholic pathway, everything here. Muscle, alcohol formation, all that is mentioned here. That's all. Now, students. Now we will learn something called as different types of enzymes. Different type of enzymes. Okay, different type of enzymes. We have something called as very simple enzymes. Very simple life they have. Simple life. Okay. Simple enzyme consists of only amino acid. Only protein part is there. Okay. Now, students, tell your physics staff. There is no physics staff and all. There is physics sir is there. Our physics sir is there. That is our Shreyas sir. There is no physics staff and all. Oh my God. Fancy word staff and all is not there. Physics sir is there. That is our Shreyas sir. That's all I know. Physics staff and all, no one is there. And I can't tell Shreyas sir. Shreyas sir will throw me out somewhere. I will tell Shreyas sir, please add. Shreyas sir will like, go look at bio. Okay. Now, students, tell me your favorite superhero. Tell one favorite superhero, all of you. Like Spider-Man, Superman, Flash, Green Lantern, Deadpool. Tell one superhero, tell me. One superhero, favorite superhero, tell me. Everyone, all the boys or girls, tell me your favorite superhero. I will teach you how enzymes work with the help of superhero. Very interesting it is. Understand? Tell me, don't be shy. What is a superhero? Okay, I will take my superhero. Okay. Tell me, I will tell you my superhero, I'll tell you. Okay. My superhero, don't tell Baswa sir again. Full flattery will not work for you, okay? Don't tell Baswa sir now. Okay. I am not Bengali, I am Kannadiga. Okay. So let's take Spider-Man. Very simple, no, Spider-Man. Okay. We will take Spider-Man first. Very simple. My favorite Spider-Man always. Okay, very simple. Now, students, there is something called as conjugate enzyme. Conjugate enzyme is like your Spider-Man. Now tell me, did Spider-Man always have the superpowers? Tell me, did the Spider-Man always have the superpowers? No. After the radioactive spider bit him, he got the superpowers later on. Now tell me, before he got the superpowers, was he able to shoot the webs and climb on the building? No. Your Apo enzyme is like Spider-Man before he got powers. That is Peter Parker. Okay. So this is Apo enzyme is before the Spider-Man got his powers. That's why, like how Spider-Man did not have any powers before, your apoenzyme is biologically inactive. Biologically inactive. Does not have any powers. Your only apoenzyme cannot do any function. Cannot do any function. How Spider-Man without powers cannot climb on building and shoot webs and fight crime, your apoenzyme cannot work. Okay. Apoenzyme cannot work. Okay. But cannot work. Now if to this Spider-Man I'm giving his powers now. Like how Spider-Man is getting the powers, the power here is nothing but what? The power is nothing but your cofactor, the non-protein part. So to this apoenzyme I am adding, to this apoenzyme I am adding the cofactor. I am adding the cofactor, the superpower, the superpower. Okay. Now, once we add to the apoenzyme the cofactor, both are joined together, we end up getting something called as holoenzyme. What do we get, students? We get the holoenzyme. Holoenzyme. So remember, students, only when apoenzyme, the protein part, cofactor, non-protein part, when they join together, we get the holoenzyme, and only when we get the holoenzyme, it is complete and biologically active. Only when the Spider-Man gets his powers, he becomes the actual Spider-Man. Otherwise, it is Peter Parker. Understood, all of you? All of you understood? What is the difference now? We have apoenzyme, cofactor. Together, they are called as what? Holoenzyme. Apoenzyme plus cofactor. If we have only apoenzyme, when only apoenzyme is, it is biologically inactive. Cofactor is activating the apoenzyme. Cofactor. Together they are complete. Together they are complete. Understood all of you? The simple concept, right? Simple concept of apoenzyme and holoenzyme. Holoenzyme is whole. It is biologically active. Apoenzyme is not very active. Understood all of you? Understood all of you? Tell me. Yes or no? Quickly in the chat. I know all of you want some break now. I know all of you want some break. But before we take a break, I'll tell you what we'll be studying next. After you come back from the break, we will be learning about how enzymes work. Exactly. How does the enzymes work? See this. How does the enzymes work? What is this? What is this? All this. So we will be learning all with the help of animation. With the help of animation, we will be learning how enzymes work. What is the mechanism of action? What is the mechanism of action? What is this enzyme product complex? All of this we'll be learning. After that, I'll be teaching you something called as what is exothermic reaction, what is endothermic reaction. Then I'll be teaching you what is this graph. Then we will learn the factors affecting. Then I'll teach something called as Vmax Km value. Then we will learn the last inhibitors. Can you see? All with the help of animation, students. All with the help of animations. What is non-competitive? What is non-competitive? See, with the help of animation. Non-competitive, uncompetitive and everything. Then last is cofactor, and with that chapter is ending. Cofactor, the chapter is ending. So that is going to be the rest of the thing which we will learn after the break. Nice. Nice. Okay. So basically what I'm telling you is, I am telling you not to leave the session basically. So what I'm telling you is, do not leave the session. A lot of new concepts are going to come later on. And the most important part is going to come later on. So now we will take a 20 minutes break. All of you quickly run and go, eat your dinner and come. And after that, don't be sleepy, that's all. Okay. Don't be sleepy after that. That's all. Okay. So we will take, yes, we will learn thermodynamics also a little bit here and there. Okay. So what is the time right now? It is 8 o'clock. Huh? 8 o'clock? No. Okay, fine. Well, it's 8 o'clock. It's 8 o'clock, almost 2 minutes to 8. So exactly at 8:20 we'll start, because I want everyone to take your rest. Take your rest. Don't go. I want to go. I want to go. I am waiting on headache. Students, I have 1, 2, 3, 4, 5, 6, 7, 8 different lights falling on my face continuously. So when you face such 8 different lights and talk for 2 hours, your brain will start to melt little. Melting of brain will happen. Okay. So we will come back at 8:20. Resume at 8:20. Okay. So if you don't want to miss all of those, if you don't want to miss competitive, uncompetitive, Vmax, enzyme separate complex and everything. Come back at 8:20. I will be here. I will be teaching. Okay. So I need some rest also, right? Only human here. So I will see you at 8:20. Quickly go and eat your food and come. 8:20. Quickly go. Run, 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 run. I need to wait. I have to order the food. I have to go down, get the food. No. How will I eat the food? No one is waiting for me outside holding the food. Baswa sir, please eat the food. No one is there. Okay, so we'll come back at 8:20, all of you. So hello students, welcome back. Yes. Where is the pen? Yes, the pen is here. Okay. So I believe all of you ate something nicely and came and ready for the next class, ready for the next part of the class, right? Okay. Yes. Time up. Sony was counting the time also. Nice. Had you dinner? Yes, I ate something here and there. Okay. Swagatam. Yes, thank you so much. Now students, the next concept which you're going to learn today right now is going to be how the enzymes work, right? So how the enzymes actually work and how are they playing a role in the increasing the time. For example, before they were making only, here is a quick example. How do they do this? Basically? From 200 molecule in 1 hour to 6 lakh molecule in 1 second. How are they able to do this amazing phenomena, right? So it's very simple. You need to understand a few concepts here. I'll teach you one by one. So understand. Okay. First of all, what is happening? We have the reactant 1 and we have reactant 2. And then we have intermediate product, which I'll be telling you what is it. Then we have product here. Right. This is a simple equation. Reactant 1 plus reactant 2. And we have a transition state called as intermediate product here. And then we have product here. Now you need to understand something called as the energy level here. Energy level is very different here. The transition state or the intermediate product before the product is formed here, right? Intermediate product is formed. Now, if this is clear, understand the definition of activation energy now. Understand the definition of activation energy. What is this meaning of this activation energy? Can anyone tell me in the chat what is the meaning of activation energy? Activation energy is nothing but it is the average energy difference between the substrate and the intermediate. Between the substrate and intermediate. Okay. Now obviously students, the reactant and substrate, the reactants and the intermediate will not have same type of energy. Same type of energy will not be there, right? For example, in activation energy, what happens is, it is the energy difference is there. Now there is an energy difference. Now imagine if the energy of your intermediate is very high. The energy of the intermediate is very high, so the reactants are not becoming product very easily. Reactant is not becoming product very easily. Okay. Yes, today it will be completed. Don't worry. Today it will be completed. Okay. So what is happening here is reactants are not able to become products very easily. Why? Because the energy of the intermediate is very high. Energy to reach the intermediate is very high. So what do we do? Now imagine all of you know high jump. So in high jump you're supposed to run and go, and you're supposed to jump on a pole and land on the other side. All of you know high jump. Now imagine the pole is right here. Tell me, will it be easy to jump over it or difficult to jump over it? Tell me in the chat. Tell me. If you're trying to do high jump, you're running and coming, the pole is right here on your head. Will it be easy to jump over the pole or difficult to jump over the pole? You will tell me, sir, very difficult. Some of you will be like, no, I can jump very well. But it is difficult to jump, right? Now, same thing is happening in energy also. Since the intermediate energy is very high, reactants are not able to become product very easily. Now you tell me, if I bring the pole all the way near the knee level, or even below that, can you jump easily now? Tell me students, can you jump easily? Tell me, now can you jump easily? You'll be like, yes sir, obviously, if I just step over it also, I can jump, I can cross the hurdle now, I can jump the pole easily. The same thing is happening here. Since the energy, the average energy difference between intermediate and reactant is very high, activation energy is very high, the catalyst is doing what now? Catalyst is reducing the energy barrier. Catalyst doing what? Like how I took the bar from here to near your leg, right? Activation energy is also reduced here. Tell me, all of you understood now. So what is catalyst doing? Catalyst is basically decreasing the energy requirement. It is decreasing the energy requirement. It is decreasing the energy of intermediate. Understand? See here, we have reactant 1. See here, we have reactant, activation energy before the bond are broken, and product is here. Okay. Now what do we do now? We add the catalyst. Now add the catalyst. What will catalyst do? Catalyst will reduce the activation energy. Catalyst is reducing the activation energy. Now reactant can become product very easily. Now students, see. Catalyzed path, uncatalyzed path. Uncatalyzed, it's very tall. Right? It is very difficult to climb. But with catalyst, it is very easy. Tell me, all of you understood? See, first this is called as activation energy. Activation energy is energy difference between reactant and the intermediate. Now we are reducing the activation energy. How are we reducing it? With the help of catalyst. So activation energy is reduced. Now since the activation energy is very reduced, reactant can become product very easily now. That's all. Catalyst is reducing the activation energy. Enzyme, product we're getting very easily. So what happens here? Enzyme plus, now enzyme is added. Enzyme plus substrate plus enzyme. We have a enzyme substrate complex. Yes, we have an enzyme substrate complex. Substrate is there, see. Substrate is adding to the enzyme. And once it adds substrate enzyme complex, then it gets converted to enzyme product complex. ES becomes what? ES is becoming enzyme product complex. Now once ES becomes EP, finally the product is coming out, the enzyme is coming out separately. So we learned in the starting itself, in the reaction, the enzyme is never consumed. The enzyme is never consumed. So understand, once again I'll tell you. First we have enzyme plus, first we have reactant. Okay. Then enzyme is being added. Now enzyme is adding with substrate. We get enzyme substrate complex, transition state. Energy is very high. Now then we get what? Enzyme substrate complex gets converted to enzyme product complex, ES to EP. Then again what happens? ES and EP split open and we get product separately, enzyme separately. Enzyme is separating it out. We're getting enzyme product complex. Enzyme is product separate, enzyme is separate. Done. Lock and key model. Any difficulty here in the mechanism? Tell me students, any difficulty here? Now chemical reaction of two types. Chemical reactions are of two types, right? There is something called as endothermic reaction and exothermic reaction. But in terms of activation energy, what is the difference? Understand again the meaning of endo and exo. Okay. Now what happening in your endothermic reaction? In the case of endothermic reaction, reactants are at lower energy, products are at higher energy. So tell me, how can reactants become taller? Reactants can become product by absorbing more energy. So reactants are absorbing more energy and then they becoming products. That's why it's called as endothermic reaction. So remember, in the case of endothermic reaction, energy is required for the reactants to become products. Reactant is at lower energy, product is at higher energy. But if you come to exothermic reaction, in exothermic reaction, reactants are high temperature already. Reactants also already have energy. Reactants already have a lot of energy. So to become products, they lose the energy. That's what's called as exothermic, is release of energy. Can you see? They're releasing the energy and they're getting converted to products. That is the main difference between endothermic and exothermic process. In endothermic, reactant are less, product is more. In exothermic, reactant are more, product are less. Is that point clear to all of you? Tell me in the chat. Simple chemistry, right? Simple biology and chemistry. Yes. Now, how do enzymes be converted? It's what is given here, we learned here. To understand this, you need to study the enzyme a little bit more. We have already understood the idea of active site. The chemicals or metabolic conversions refer to reactions. The chemical in which is converted to product is called a substrate. All of you know substrate. Hence, enzyme, the protein which three-dimensional structure including an active site, converts the substrate into a product. Substrate is becoming product. Okay. Where is it? It is now understood that the substrate has to bind to the enzyme at an active site. Substrate has to bind, substrate not enzyme. Remember this is very important. The substrate has to come and bind to the enzyme. That is very important to learn. Substrate is coming and binding to the enzyme. At what site? At a specific site called as active site. Not activa gadi, active site. Okay. So enzyme is coming in, substrate is coming and binding to the active site. Within a given cleft or pocket, the substrate has to diffuse towards the active site. See, the substrate has to diffuse towards the active site. Substrate has to come. It's coming and binding now, substrate. There is thus an obligatory formation of enzyme substrate complex. The E for enzyme. This complex formation is a transient phenomena, means very temporary. During the state where the substrate is bound to the enzyme active site, a new structure of the substrate called as transient state structure is formed. Temporary site again. Okay. Very soon after the expected bond breaking and making is completed, the product is released from the active site. Once product is made. In other words, the structure of substrate gets transformed. See, what is transformed? In other words, the structure of substrate, enzyme is not modifying. Enzyme is remaining unchanged. It is not getting confused. The substrate is becoming, right? The substrate is turning into a different structure. See, the substrate gets transformed into structure of product. This can also be asked as the AR question. AR question. Clear all of you? The substrate is getting modified now, not the enzyme. The few points that I'm telling you here are very important points. Okay. The path of this transformation must go through a so-called transition state structure. There could be many more alternative structure states between the stable substrate and the product. Implicit in this statement is the fact that all the other intermediate structural states are unstable and temporary. Stability is something related to energy status of the molecule or the structure. Hence, when we look at this point of this pictorial through the graph, if you look at the graph here, this is the substrate, this is the product. Here we have the transition state in which we have enzyme substrate complex. Then what do? Once we add the enzyme is reducing the activation energy. See, activation energy with the enzyme. So since activation energy has been decreased, substrate can now easily form into products. That is what is happening here. Concept of energy. The y-axis represents the potential energy here. Potential energy, y-axis. The x-axis represents the progression of structural formation or state through transition state. You would notice two things. The energy level difference between substrate and the product. If product is at lower level than substrate, if product is lower level, the reaction is exothermic. We learned this right now, right? If product is lower level, it is exothermic in nature. One need to see, I'll repeat again, students. If product is at lower level, it is exothermic. Okay. Low exothermic. One need not supply energy by heating in order to form the product. However, whether it is exothermic or spontaneous reaction or an endothermic reaction or energy required reaction, the substrate has to go through a much higher energy state or transition state. I told you, transition state has higher energy. Transition state has a higher energy, right? The difference in, this is the line I told you here, the difference in the average energy content of substrate that from the transition state is called as the activation energy. The energy difference between the substrate and the transition, that is called as the activation energy. This same activation energy needs to be reduced by the enzyme. Students, are you getting the feel of it? Are you understanding the entire beautiful lines of NCERT here, given here? Beautiful lines of NCERT, all of you. Yes, all of you understanding? Yes. Transition state is always high. Transition state is always higher. So to reach there, it has to spend more energy, was to acquire more energy, and transition state is decreased by enzyme, so it is becoming product again. This is the example of endothermic reaction. See, product is low, say exothermic reaction. Okay, exothermic reaction. Beautiful concept. The enzyme eventually brings down its energy barrier, making the transition of S to P more easily. S to P more easily. So that is how enzymes work, students. Enzymes work. There is an activation energy, the energy difference between the reactants and the transition state. Activation energy is decreased by the enzyme. Substrate is becoming product very easily. Beautiful lines of NCERT. Now, nature of enzyme. In each enzyme, E has a substrate S binding to its site. Students, always remember, I've always told you now, always remember the substrate will diffuse and come and bind. It is never, enzyme is never going bind. Enzyme is chilling on its own. The substrate will bind to the enzyme. Why? Because the enzyme has the active site. Now you tell me, if you have money, if you have food, your friend has food. If you have biryani, okay, let's say Tanuja has biryani in her house and she's calling Ravindrani. Will Ravindrani come and eat biryani? Obviously, she'll like free biryani, she'll go. Or Tanuja will take the biryani and go to her house. No. Tanuja has the biryani, she is the boss. She will hold the biryani in her house. She will wait for others to come to her house. Understood? Very simple. Substitutes will bind to the enzyme because enzyme has the active site. Okay. Now, highly reactive enzyme substrate complex ES is produced. This complex is short-lived and dissociates into product and the unchanged enzyme with intermediate formation of enzyme product complex. See, we have enzyme substrate, complex, EP complex, EPN separated out. EPS separated out. Okay. Now all of you understood, right? Sumi, you understood? Enzyme substrate complex. An enzyme will always stay aram se. Why? Because it has the active site. It knows PV will come to me because I have active site. Your substrate will go and bind there. Substrate will go and bind there. Okay. Now, what is happening? First, substrate bind to the active site of the enzyme. So always it's telling you, substrate binds to the active site of the enzyme, fitting into the active site. The binding of substrate induces enzyme to alter its shape, fitting more tightly around the substrate. So first of all, imagine Ravindrani is going to Tanuja's house. As soon as she goes, will she start eating completely? No. She'll become comfortable first. Remove the chappal, remove the jacket, then nicely wash the hands, then sit for eating. Here also, the binding of the substrate induces the enzyme to alter its shape. So first, initially what happens? Initially, it is aram se joined. The enzyme is joining very loosely. After some time, after some changing shape and all, the enzyme is completely strongly binding with the substrate. First loosely bound, then tightly bound. Loose to tight, loose to tight. Okay. Now, the active site of the enzyme now is closely proximity of the substrate. Break the chemical bonds of the substrate and the new enzyme product complex is formed. I'll repeat again. The active site of the enzyme now is in close proximity of the substrate. So active site, substrate, they're very close to each other. Now what will happen? The Ravindra will take the food, go to the mouth. What has happened now? So finally the enzyme substrate complex will break into enzyme product complex. enzyme product complex. What happens next? The enzyme releases the product of the reaction and the free enzyme is ready to bind to the another molecule of the enzyme. So what happens? Once Ravindra is done, Ravindra will go back home. Next, one more person will come and start eating in the house. Understood? So all the substrate has to come to the active site only. Understood? Understood all of you students? This is very easy to understand. It's only a matter of, if you just read it one more time, after I am done with my class, watch the session one more time, you will understand it very beautifully. Okay. It's a good story written here. Enzyme complex. Now students, there are many factors. There are many factors which inhibit or promote enzyme. Right? Obviously, there are many factors. Now, there are many factors to eating biryani. Right? We'll go to biryani today, full biryani mode today. Okay. Because I can't eat in the house, obviously, that's why. Only dreaming of biryani, not eating of biryani. Okay. Now imagine if you're not hungry, will you eat biryani? No. If very hungry, you'll eat more. If biryani is made really well, you'll eat even more. With biryani, there is chicken kebab also, you'll eat even more. Right? So depending upon the factors, how much you're hungry, how tasty is the biryani, what are the sides, do you have juice after that? Right? How much you have to travel for it? Is it made in your house? There are many factors to it, right? Similarly, for an enzyme to work, there are many factors involved. Okay. Many factors involved. There is, for example, temperature, pH, substrate concentration, enzyme concentration, and inhibitors. There are multiple factors affecting the activity of enzyme. For example, temperature. Now tell me students, all of us know your enzymes are basically what? Enzymes are basically proteins. High temperature, enzyme does not work. Low temperature, enzyme does not work. So enzyme will only work well when there is optimum temperature. See, narrow range of temperature which enzymes are active. Optimum temperature at which the enzymes is maximum activity. High temperature, denaturation. Low temperature, inactivation. Same goes for pH. There is a narrow pH. For example, pepsin will work at 1 pH. Salivary amylase will work at 5.5 pH. Alkaline will work at 9 pH. So every single enzyme has an optimum pH at which they work. If the pH is more or less, enzyme does not work. Very choosy enzyme is very picky. Little bit also, if you left and right if you make, enzyme does not function. Understood? So there is something called as optimum pH also. Optimum pH. Now, what about substrate concentration? Understand? Very easy now again. Understand substrate concentration. Now you tell me, all of you look here. Here we have substrate concentration in the x-axis. Substrate concentration. Here we have reaction. How fast the reaction is happening. Now, what are we doing here? Here we have enzyme. And we have substrate here. So can you see in the first block? There is a very low substrate. So the reaction is a little bit slower. Because not all active sites are filled. All of you can see, right? Not all active sites are filled here. Some of the active sites are empty active sites. Why? Because there is less substrate. There is very less substrate here. Okay. Now what if we increase the substrate concentration? Will the reaction increase? Yes. So increase in substrate concentration is directly proportional to rate of reaction. So as you increase the substrate, reaction is also increasing. Okay. Understand? So as the substrate is increasing, rate also is increasing. Can you see? So can you see at this second block, majority of substrate are being completely filled. So majority of the active sites are filled here. Can you see? Majority of the active sites are filled here now. Yes. Now what happens once? Now again if I'm increasing students, the substrate concentration, will the reaction again increase more? No. Why no? Because at one point, all enzymes or active site, all active sites are already filled. At one point, all active sites are already filled. So there is nothing left. There are no enzymes. If 10 people come to Tanuja's house, 20 people come, but biryani is made only for 5 people, can she still keep on feeding everyone? No. So at one point, students, at one point, how many substrate do you add? How much ever substrate do you add, the reaction will not increase, the reaction will become stagnant. Reaction will become stagnant. All of you understood now? Tell me yes or no? So initially what is happening? As you increase the substrate, reaction is also increasing. But at one point, all the enzymes, all the active sites are already filled. It doesn't matter how much ever you increase. It doesn't matter how much ever substrate you increase, the reaction will be constant. The reaction will be constant. Clear, all of you? Clear? Clear? Yes? Beautiful, beautiful. This is called as substrate concentration. The same goes for enzyme concentration also. The same goes for enzyme concentration also. Now students, understand what is Vmax. Understand what is Vmax here. What is this Vmax? Vmax, understand. Vmax is nothing but maximum velocity at which all active sites are filled. All active sites are filled. So eventually all the active sites will fill. Eventually all active sites will fill. So Vmax is nothing but at a maximum velocity at which all active sites are filled. Now students, it becomes very difficult to find Vmax. It becomes very difficult to find all active sites, to count, to see when are they filled. So we can't really find Vmax, but can we find half Vmax? Yes. What is half Vmax? Half of the velocity at which all active sites are filled. That's all. What is half Vmax? Sorry, not half velocity. Velocity at which half Vmax. Velocity at which half of the active sites are filled. Active sites are filled. Not half velocity. Velocity at which half of the active sites are filled. Since you can't find all the active sites, we are only finding the half of the active sites. Students, write down this. This is very important. Write down this, very, very important. So what is Vmax? Maximum velocity at which all active sites are completely filled. What is half Vmax? Velocity at which half of the active sites. Since we can't find all of them, we are finding some of them. Half of the active sites are completely filled here. Half of the active sites. Yes. Now you tell me, if active sites are being filled, some amount of substrate needs to be coming and joining there. So what is the meaning of active sites being filled? The meaning of active sites being filled is that substrate is joining there. So students, at half Vmax, can I tell some amount of substrate is joining the active site? All of you tell me, agree with me in the chat. At half Vmax, where half of the active sites are filled, some amount of substrate has to be joined there. Yes. So the amount of substrate, students, or the concentration of substrate, concentration of substrate concentration of substrate at half Vmax, at half Vmax is called as Km value. It is called as Km value. This is half Vmax. Clear all of you? Clear all of you? Tell me. So at half Vmax, right, can you see? At half Vmax, the amount of concentration of Km which is used. At half Vmax, right here, amount of Km which is used, amount of substrate which is used is called as Km value, also called as Michaelis constant. Michaelis constant is not given in syllabus, but remember. So this is Km value. Concentration of substrate at half Vmax. So at half Vmax, how much of substrate is used, that is called as Km value. It is called as Km value. Okay. Now understand, students. Let's read what is given in NCERT now. Let's read what is given in NCERT. Okay. The activity of enzyme can be affected by change in conditions. All of you. See, temperature and pH. Enzyme generally function at a narrow temperature and pH. Each enzyme shows its highest activity at a particular temperature and pH. That is called as optimum pH. Called as optimum pH and optimum temperature. Activity declines both below and above the optimum value. Go above the optimum value, decrease. Below the optimum value, decrease again. Low temperature preserves the enzymes at temporary arrest. Inactive stage, high temperature completely destroys the enzyme. Denaturation. Concentration of substrate, we learned this, you learned this already. You learned this already. With the increase in substrate concentration, the velocity of enzymatic action rises at first. See, initially it is raising. Okay. The reaction ultimately reaches the maximum velocity called as Vmax. Vmax is the maximum velocity at which all the active sites are completely filled, which is not exceeded by any further raise in concentration. So after once Vmax is reached, how much amount of substrate you add? How much amount of substrate you add? There cannot be more increase in reaction. We learned here, right? How much ever after one point, this is the Vmax. Once we achieve the Vmax, how much ever substrate you add, the rate will not increase. The rate of reaction will not increase. Okay. This is because the enzyme molecules are fewer. Now enzyme is fewer, obviously. Enzyme is very less. Biryani is very less. Are very fewer than the substrate molecules. And after saturation of these molecules, that is, once all the substrate has been used, there is no free enzyme molecule to bind. Obviously, there is no active site again available with the addition of substrate. So how much ever substrate you add, reaction will not increase any further. The activity of enzyme is also sensitive to presence of specific chemical that binds to the enzyme. When binding to the chemical sheds off the enzyme activity, the process is called as inhibition and the chemical is called as inhibitor. So what is inhibitor doing? Inhibitor is the thief. Inhibitor now will silently go. It will silently go and join the active site. First of all, active sites were very less. Now instead of substrate coming and joining, the thief inhibitor will go silently and join the active site. That is called inhibitor. That is called as inhibitor, the thief, the kalla. It is going and joining the active sites. Now we will learn different types. Understand? Ready, students? Ready to learn the last two concepts left? Last two concepts we can, after you can go home. After you can go home, do what you are already in the house. So sleep off after that. Okay. Ready, students? Ready to learn the inhibitors now? Ready, all of you? Yes, thief is silently stealing the biryani. Okay. Double acting of a hero. What double acting you're telling? Huh? I should become an actor now. Now can we start, students? Inhibitors now. Inhibitors, very beautiful, very simple concept. Understand with the help of the reactions. Okay. Now, inhibitor. chemical that shuts off the enzyme activity. In other words, inhibitor inhibit the enzyme activity. See, this is the enzyme. See, substrate was supposed to join. Instead of substrate joining into the active site, your inhibitor coming from the back door and it is joining. You can see inhibitor is coming and joining here. I want kalati from back. Inhibitor is coming and joining here. Can you see? And if inhibitor is joining here, substrate cannot join anymore because there is no food left. Right? There is no food left. Now, understand there are three types: competitive, non-competitive, and uncompetitive. Now, first learn what's called. My lace came out. 1 second. My lace came out. Shoelace. I stamped on my lace. I don't want to fall down. That's why. Let me tie my lace. Oops. What is competitive inhibitor now? Competitive inhibitor. Understand. Your competitive inhibitor will look exactly like the substrate. There are instead of Ravindrani coming and eating the biryani, the clone of Ravindrani is coming and eating the biryani now. Understand? Your competitive inhibitor resembles the substrate. It will look just like the substrate and compete with the substrate for the active site. See? So, instead of students, understand. Here we have the, instead of complex enzyme inhibitor complex is forming now. Instead of enzyme substrate complex. Instead of enzyme substrate complex, what do we have now? We have enzyme inhibitor complex. Enzyme inhibitor complex we have. Enzyme inhibitor complex. Understood all of you? Understood? Instead of enzyme substrate complex, we have enzyme inhibitor complex. Why? Because your inhibitor looks just like your substrate. You can't tell only which is inhibitor, which is substrate. No, same to same. Pakka copy it is. Same to same it is. You can't tell the difference between which is the inhibitor and which is a substrate. Which is a substrate. Here you have a famous example. Here you have a famous example, that is succinate. Succinate is getting converted to fumarate. Succinate is getting converted to fumarate. Okay. The enzyme here is, the enzyme which works here is called as succinate dehydrogenase. Succinate dehydrogenase. Now what happens is, this is the enzyme. Now we have an inhibitor. Inhibitor is called as malonate. Malonate. Okay, what is happening now? Malonate is coming and joining here. One thing, students. What is happening now? Malonate is coming and joining. Instead of, it is coming and in the place of succinate, malonate is forming with enzyme and we are not getting the product. Understand all of you? So, clear? Tell me, this malonate is forming a bond with enzyme and we are not getting the fumarate. Understood all of you? So, malonate is the inhibitor for the succinyl dehydrogenase enzyme. Succinyl dehydrogenase enzyme is forming a bond with malonate and we are not getting the fumarate. Beautiful. Competitive inhibitors. Okay. Now what about non-competitive? What exactly happens in non-competitive? Understand. Non-competitive. So your non-competitive does not want to fight. So non-competitive will do what? It binds at the site other than the active site. See, here we have inhibitor, substrate. So instead of it is able to go and join here, instead of substrate able to join here, inhibitor is joining and it is changing the shape of the enzyme. That is, inhibitor will bind with somewhere in the other part and it will completely change the shape of the enzyme. Now the enzyme shape is only changed. Now can the substrate come and join? No. That is, change the shape of the active site such that substrate is no longer able to bind. can bind to free enzyme or enzyme should complex directly. See, it is binding the free enzyme, change the shape, it cannot bind anymore. Non-competitive. So remember, this point is very important, last point is very important. It can bind to free enzyme or enzyme substrate complex. To a complex also it can bind. Other than active, like behind the back it is coming, behind the back it is coming. See here, it's coming from the behind the back and joining here, changing. Your substrate cannot join now. Substrate cannot join. Okay. Okay. Clear? This is non-competitive. Now what about uncompetitive? Uncompetitive, very simple. Binds to the enzyme at the site other than the active site. It cannot bind to free enzyme. It cannot bind to free enzyme. So what does it do? So it will wait. It will nicely wait for a substrate to attach first. See, it cannot bind first of all. So substrate will attach. Then it will attach and kick it out. Understood? So uncompetitive is very simple. Uncompetitive will wait. You have seen this cat waiting for everyone to go. They'll go to drink the milk silently. So your inhibitor is waiting for the complex to be formed. Once the complex is being formed, then only inhibitor will attack. Can't attack first. So it will join, then it will attack and kick it out. This is called as uncompetitive. Uncompetitive. Understood, all of you? Tell me, is it difficult? Is it difficult to understand all this? Very easy, right? So non-competitive, uncompetitive and competitive inhibitors. 3 different types of inhibitors. Sir, do you have live tomorrow? Yes, I have live tomorrow. I take class every day. You think I'm taking rest? There's no rest. Since your exam is coming near, no rest. Okay. Now, that was inhibitors. This is your Vmax, half Vmax, Km value. Km value. Optimum pH, optimum temperature here. Now we come towards the one of the last few topics for today's students. Ready? Ready, students? Tell me. Last few topics for today's class. Last few topics for today's class. Okay. Students, do you remember the superpower which I gave to Spider-Man? What was it called as? Can anyone tell me? Tell me, students. What did we call that superpower of Spider-Man? We had apoenzyme. Something was adding to the apoenzyme. Can anyone tell me? This is your apoenzyme. Something is adding and making it holoenzyme. What was that? That was nothing but your cofactor. Do you remember? Cofactor is adding with the apoenzyme and we're getting the holoenzyme. Apofactor. Yes? A cofactor, remember? So here also we have different types of cofactors. We have different types of cofactors. Okay? We have something called as organic cofactor and inorganic cofactor. We have something called as organic cofactor and inorganic cofactor. Inorganic. Under inorganic cofactor, we only have metal ions. Metal ions are giving the power to the holoenzyme, to the apoenzyme. Now under organic part, we have two things. We have something called as prosthetic group, also something called as coenzyme. Prosthetic group as well as coenzyme. Clear, all of you? Prosthetic group and coenzyme. Two things are here. Okay. Metal ion, prosthetic group and cofactor. What is the difference between all 3 of them? Understand. Let's read NCERT. It's very clearly given in NCERT actually. Cofactor. See this. First of all, tell me in the chat. Your apoenzyme was protein part. Cofactor is non-protein part. Right? Cofactor is non-protein part. The enzymes are composed of one or more several polypeptide chains. However, there are a number of cases in which non-protein constituents are called as cofactors. Non-protein cofactors, okay, are bound to the enzyme to make the enzyme catalytically active. Okay. In these instances, now what is the meaning of catalytic active? It is providing energy, providing powers to the apoenzyme to make it holoenzyme, biologically active. In these instances, a protein portion of the enzyme is called as the apoenzyme. See, coming here, apoenzyme is the protein part. 3 kinds of cofactors may be identified: prosthetic, coenzyme, metal ion. 3 different types of cofactor. Understand. The first one is called as prosthetic. Okay. Prosthetic group are organic compounds. See, organic, told you already, organic group, and are distinguished from the other cofactors in that they are tightly bound. So prosthetic groups are what? Tightly bound. Tightly bound. Okay. To the apoenzyme. Where? To the apoenzyme. Okay. For example, peroxidase and catalase. Which catalyze? Peroxidase and catalase, the 2 enzymes, and catalase which catalyze the breakdown of hydrogen peroxide into water and oxygen. So what do we have, students? H2O2 is getting converted into water plus oxygen. Yes, water plus oxygen. The enzyme here is what? Enzyme here is peroxidase and catalase. Now this peroxidase and catalase, they cannot work on their own. They need that cofactor. Okay. What is the cofactor? The catalyze the breakdown. The heme is the prosthetic group, which is the part of the active side of the enzyme. So the power is given by whom? The power is given by the heme group. So this is the example of prosthetic group, tightly bound to the apoenzyme. Example is hydrogen peroxide getting converted to H2O2. Peroxidase, catalase, prosthetic group. The example of prosthetic group is nothing but your heme group. Clear, all of you? Heme is the prosthetic group. Heme. Okay. The first one is done. Here. Till here is done. The second one is called as coenzyme. Coenzyme are also organic compounds. See, they're also organic. Both are organic in nature. But their association with the apoenzyme is only transient. So, unlike the prosthetic group, coenzyme only bind what? Coenzymes are bound only transient. Means very short form or loosely bound or for a short period of time. Transient. Okay, transiently bound. Usually occurring during the course of catalysis. Furthermore, coenzymes serve as cofactors in a number of different enzyme catalyzed reactions. The essential chemical components of many coenzyme and vitamins. Example, coenzyme nicotinamide adenine dinucleotide. The example here is, example of your cofactor coenzyme is going to be what? NAD and NADPH. They also contain vitamin niacin. They are the example of coenzyme. They also have something called as vitamin niacin. They have niacin because niacin is, students remember, niacin is not the coenzyme here. Niacin is not the coenzyme. Niacin is a part of the structure of NADP. Your NAD and NADPH are the coenzymes. They are the coenzymes. Now, last part. Third one is called as metal ion. It's called as the metal ion. A number of enzymes require metal ions for their activity, which form the coordination bond with the side chain of their active site, and at the same time forms one or more coordination bond with the substrate. Example is the zinc. Zinc is the cofactor of the proteolytic enzyme carboxypeptidase. For the enzyme carboxypeptidase, students, for the enzyme carboxypeptidase to work, it needs a metal ion that is nothing but the zinc. Carboxypeptidase. Example for your metal ion, carboxypeptidase. Now, catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that they play a crucial role in the catalytic activity of the enzyme. So if you remove the cofactor, the enzyme cannot work anymore. It cannot work anymore. The last part of today's class, the last part, which I'll be finishing in the next 5 minutes, is starting right now. After this, I have few questions. Let's solve a few questions also. Okay, don't run anywhere. Solve the questions. Okay. Now, classification of enzymes. Thousands of enzymes have been discovered, isolated and studied. Most of these enzymes have been classified into different groups based on the type of reaction they catalyze. Enzymes are divided into 6 classes. 6 classes with 4 to 5 subclasses, 4 to 13 subclasses. First one is something called as oxidoreductase or dehydrogenase. The enzyme which catalyzes the oxidoreduction between two substrates. Basically, reduction, oxidation, oxidoreduction, redox reaction. Redox reaction is happening. Okay, redox reaction. Okay. Now transferase. Enzyme catalyzes the transfer of group of B. So here transfer is happening. G is being separated out. Other than the hydrogen, between the pair of substrate called transferase. Then we have hydrolase. This is very simple, students. Hydrolase. See, I'll give you example of transferase. Okay. A plus B is there. Okay. A plus B. We have AB here, students. We have AB plus C. What will happen? We'll have A, C bond and B is removed out. B is removed out. This is a transferase. Simple example of transferase. Okay. Now hydrolase. Hydrolase. Remember, students, in hydrolase only we have the amylose, pepsin and everything. Okay. In enzyme catalyze the hydrolase of ester, ether, peptide, glycosidic, aldehyde, C halides and PN bonds. Hydrolase enzyme. Okay. Lyase. Lyase is breaking the bond with breaking the bond, making a double bond. Enzyme that catalyzes the removal of group from substrate by mechanism other than the hydrolysis is called as hydrolysis, leaving a double bond. So hydrolase will break the hydrogen bond. Lyase will also break it while making a double bond. See, it will make a double bond. It will make a double bond here. Isomerase. Include all the enzymes. Students, remember isomerase. Glucose 6-phosphate to fructose 6-phosphate. Glucose 6-phosphate to fructose 6-phosphate. Isomerase enzyme. Right? Isomerase. You also learn isomerase in your Krebs cycle also. Krebs cycle also is there. Okay. Ligase. Enzyme which joins. Obviously, ligase is joining. Ligase is joining here. It joins two compounds. Time starts now. Let's see who answers this question first. Let's see who answers this question first now. Time starts now. Go. Let's see who answers this question first. The inhibition of succinate dehydrogenase by the enzyme malonate is a classical example of inhibition of succinate dehydrogenase enzyme by malonate. So malonate is coming and joining succinate dehydrogenase, inhibiting it. It is an example of what? I told you. It is an example of competitive inhibitor. Competitive inhibitor. The cofactor of enzyme carboxypeptidase. The cofactor of enzyme carboxypeptidase. We just now learned carboxypeptidase. Carboxypeptidase is the answer. Which is the answer? Niacin, flavin, heme, zinc. Carboxypeptidase. Friends, I'll tell you. Learn this one particular paragraph very well. Carboxypeptidase, zinc. NADP for your coenzyme. Here we had heme for your prosthetic. So what is the answer? What does that tell me? What is the answer now? Obviously, zinc is the answer. Zinc is the answer. In a reaction catalyzed by an enzyme, which of the following statement is correct? Which statement is correct now? The enzyme decreases the activation energy for formation of transition state. Enzyme makes a transition state from substrate to product more difficult. Enzyme increases the activation energy. Enzyme substrate complex formed during the reaction lasts for a very long time. Which is the right answer? Enzyme decreases the activation energy, right? Enzyme decreases the activation energy, or enzyme makes a transition. Enzyme make transition from substrate to product more difficult. No, it does not make difficult. It makes much more easier. A. Yes, absolutely A. It is making it much more easier, decreasing the activation energy, right? Decreasing the activation energy. Which of the following statements regarding enzyme inhibitor is incorrect? Incorrect. Competitive inhibitors is seen when a substrate competes with an enzyme for binding to a inhibitor protein. A competitive inhibitor is seen when the substrate and inhibitor for the active site of the enzyme. Non-competitive inhibitor of an enzyme can overcome by adding large amount of substrate. Non-competitive inhibitors often bind to the enzyme irreversibly. Very good question. Very good question. Which is correct. Very good, very easy question, very easy question. Think. This is not NEET PYQ, which I have got this question from so many different places to make. Yes, exactly. Very simple, right, students? Your competitive inhibitor is what? Competitive inhibitor is nothing but when the substrate and inhibitor compete for the active site, right? Both are competing for the active site. Why? Because both are same to same. Both are same to same. So they are dividing for active site. That's all. It may look confusing at first, but it is very easy. Okay, very easy. Now tell me, once you know the answer, try reading the answer. It makes much more sense now, right? Look at this question now. Malonate inhibits. Okay, this is a 1 second. Malonate inhibits. Oh, more question is there. Okay. Malonate inhibits the growth of pathogenic bacteria by inhibiting the activity of. Wait, did I miss this line of NCERT somewhere? Malonate. Okay, that line is removed. I don't know why line is removed here. All of you know this answer, right? Malonate inhibits the activity by which one? By stopping the activity of succinyl dehydrogenase. Competitive inhibitor. Next question. Low temperature preserves the enzymes in a temporary state, inactivate state, where high temperature destroys the enzymatic activity because proteins are denatured by heat. Okay. When an inhibitor closely resembles the substrate in its molecular structure and inhibits the activity, it is known as competitive inhibitor. It is known as competitive inhibitor. What is the answer? Yes, Sujan, there is a connection between students. Let me tell you, learn biomolecules right now. Your chemistry will become very easy. Students, read the first statement. In low temperature, the enzyme is temporarily inactivated. Low temperature, enzyme is inactivated. Right. High temperature, enzyme is easily destroyed. Is it wrong? Correct? Is it correct or wrong? Tell me. The statement number 1. Does it make sense? In high temperature, enzyme is denaturing. Low temperature, it is inactivated. Makes sense. Yes. So both are correct. Absolutely, both are correct then. All of you understood? Both are correct. The first one is also correct. Why? Low temperature preserving the enzyme. In temporary state, low temperature becomes preserved. This is your homework question. And I want it in the comments. Students, 3 hours 20 minutes you have taken the class. 3 hours 20 minutes the class has been done. NEET 2024 question, I want in the comments. Okay. So also let me know in the comment section how was today's class, because it's very important. Feedback is important, right? Feedback is very important. So let me know in the comment section how was today's biomolecules. And with that, see, this question is a little difficult towards the end because I found them separately. Okay. Go try solving NEET PYQs. Okay, you will feel super confident, and the chapter is done for all of you now. It's only a matter of revision. Try revising once or twice. Biomolecules will become very easy for you. Okay. So, and let me tell you students one more thing before I go. how many of you have not registered for NEET till now? Raise your hands. How many of you are not registered for NEET till now? So quickly do it, because all of you are aware that NEET exam is a scholarship exam, doesn't require any fees, and you can simply go join it, write it, and if you win, you can get a PlayStation, you can get an iPhone, iPad, scholarship in our paid batches, everything is there. So write the NEET exam. Okay. Another question you want? Okay. See this question. All of you see this question. Simple question. Look at it. I will tell you the answer now only. Okay. The question is very simple. The question is very simple. Tell me which is a better enzyme. Which is a better enzyme? A or B? Which is a better enzyme? Time starts now, go. Which is a better enzyme? Is it A or B? Which is a better enzyme? Sir, we only know organic chemistry very well. Could you very well? Only we could know this chapter well. Huh? Sujan, I don't understand. I'll teach you, students. I'll teach you now. Your A enzyme is very better. A enzyme is better. Why? Because enzyme A is reaching half max with only around 1% of only one usage of nutrients or substrate. So your enzyme A requires less substrate. Enzyme A requires less substrate for reaching half Vmax. While your enzyme V is consuming a lot of substrate to reaching half Vmax. So it is a rule always. Good enzyme will have less Km value. Good enzyme will have less Km value. I already told you the answer, but that is. But no, this is fine. Tell me answer this question only. This question only you tell me the answer. If you know, you know. If you don't know, you don't know, obviously. So thank you so much, students, and take care, all of you. I will see you tomorrow, day after, someday. But before I go, I want everyone to quickly mark your attendance now only. If I don't tell, all of you don't do only. Some weird thing all of you have. Okay. So if you go here, students, if you go right here, right now, my face is seen here, but if you go to Vedantu NEET English channel, and if you go into live, and if you scroll down, do you see this? Go here and quickly smash the like button, all of you. Smash that like button. So part 1 we will be doing photo respiration in plants. Part 1 after biomolecules. After biomolecules. So with that being said, thank you so much, students, and I will see you in the next class. Today's class was really fun for me, and hopefully it was fun for you also, and you learned a lot. And congratulations on staying up till more than 3 hours. right? So bye bye, take care all of you, and have a great night ahead. Bye bye.